It's my great pleasure to be able to introduce and kick off this year's Galaxy Community. What an exciting time to be here. I'm really excited about the fantastic program we have this week, all the talks, the presentations, all the trainings, all the activities, be able to see this wonderful city, this wonderful town, bring together this wonderful. This is absolutely one of my favorite conferences of the years. I mean, we spent, I don't know, more than a year planning this one. There's just so much exciting things that are going to be happening this week. It's just incredible to be able to celebrate Galaxy and celebrate the Galaxy community. It's all about bringing people together. Before diving into the program, there's a bunch of people I really wanted to thank and acknowledge for all their hard work that has been going into this whole program for the past year to make it so successful. So first of all, many thanks to Masaryk University, the Elixir Czech Republic, and the city of Brno for hosting it. Let's give everyone a huge round of applause. I think this is an absolutely fabulous venue. What a great room to bring everyone together. What a great city to host everyone. There's so much activity with Galaxy going on here. This was just a really central location. We'd like to see that this environment here just be promoted through this. Next of all, I'd like to thank the overall GCC organizing committee. So this is Bjorn, Ennis, Hulls, Jen, Natalie Whitaker, for their tremendous hard work going on behind the scenes to put together the program. Let's thank them. I think between them, that's probably been, I don't know, hundreds, thousands of hours on the phone, on Zoom, communicating, just making sure everything is sort of, organized. And then, right in partnership with them, there was a set of committees for trainings, for the scientific program, for all the activities, for all the CoFest activities and so forth. So there's, I don't know, 100 odd people that have been behind the scenes working to kind of get everything organized. So let's thank them. Then, of course, beyond sort of the organization, beyond all the sort of people behind the scenes, thanks to all of you for coming here. I think we've had people traveling, I don't know, hundreds, thousands of miles from all corners of the earth to be here. It's about bringing the Galaxy community together. A conference is only successful when people are actually here to present all their work. So thank you for traveling. It's been awesome. And I look forward to seeing you all. Next, I'd like to thank all of the speakers this week, all of the poster presenters, all of the trainers, and really just everyone for your participation, for your sharing. This is all about bringing the community together, and I really thank you in advance for sharing all of your ideas and just being so open with your time and so open with building this community spirit. And then finally, beyond the people in this room, there's a bunch of people on Zoom. And then just very broadly, thanks to the entire Galaxy community for all your contributions throughout the year, throughout the entire years. Right, this project is only successful by through all the contributions of everyone, even from a single line change to a single training to a single activity to just running a single job. That's where it all starts. Obviously, we want people to be as engaged as much as we possibly can, but it all starts with that first contribution. So thanks to the whole world for everything. Before kind of diving into the talks this morning, there's a couple other short presentations. So first up, I think is Michelle is going to kick us off with a kind of a quick review of the code. Michelle.
Okay, so the okay, so where did we come from last year when we were in Brisbane? Yes, we came from 20 countries, over 500,000 kilometers traveled. And we represent a very, very diverse group. So as part of the Code of Conduct Committee, I have, yes, with great power comes great responsibility in terms of making everyone feel welcome and knowing a little bit about Code of Conduct and what it's about. So it's a living document. It does change on occasion, but it's managed by the Code of Conduct team. We meet annually about, semi-annually now about. And the principles are, for everyone's benefit, pretty basic: be friendly and patient, welcoming, be considerate, respectful, try to understand why we disagree, and be careful in that we choose. And these were all created by members from all of us. Okay. So I don't think we have stickers this year, but it is good to just be mindful in terms of people who might have different levels of comfort in terms of proximity or various, and to respect those boundaries. So as part of the code of conduct board, then one of the things, or I should say, one of the responsibilities we have is to really kind of protect all of you and make sure things run smoothly, continue to run smoothly. And every year we do file an incident report, which we try to make transparent. And so for this last year, 2023, things went very, very well for the most part. And we didn't have any incidents reported to us. Historically, in the last years doing this, it's only been one at an outside event. But you are welcome to us. We do have annual updates, which by Galaxy. Okay. Yeah. So if you see any of us while here and you experience anything that is a code of conduct issue, please feel free to reach out to one of us. Right now, there are 4 of us. We lost a member earlier this year who's off somewhere else. And along those lines, we are looking to get new candidates. So we have a new candidate form. If you have experience being on a code of conduct committee or even something similar, we'd love to have you. And actually, how we got formed was a little bit happenstance, should I say. So many of us will be filling this out for ourselves to make sure that we also will continue to be a part of the community and represent in the way that it needs to be. Let's see. Yep. Special thanks to our host country and organizers, and thank you all for coming.
Thank you, Michelle. Okay, next up, Helga's going to give a few opening remarks and then introduce the first keynote speaker.
Thank you so much. Yeah, first of all, thank you very much, Mike, for this great introduction. And I would really like to welcome all of you to this year's Galaxy Community Conference here in Brno. Brno might not be your first holiday destination and might not have the appeal of Prague or Vienna, but I think it offers great opportunity to just relax after the conference. We'll have beautiful weather during the time here, and you can really see life in the city, and I'll hope you'll get an idea of how it is to live here in this beautiful city. So Brno is also the place where Gregor Mendel actually worked and did his foundations on genetics, which is the discipline which inevitably somewhat actually caused this project, because they had so much data, they didn't know how to deal with it. So we have Galaxy. And genomics is actually not the only discipline or the only thing that actually influences health. The environment we live in, the air we breathe, what we eat, what we do, what we are exposed to, has an impact on us, our physical and mental health. And it's this gene versus environment interaction which inspired the conception of the exposome, the totality of exposures of an individual during their lifetime. And on that topic, it is really my pleasure to introduce our first keynote speaker, Jana Klanova, Director of the Research Center for Toxic Compounds in the Environment. She's been a professor for environmental health sciences since over a decade, and as of 2021, she's also the director of the newly established IRENE-RI. Jana Klanova knows the importance of translating science into policy and of communicating results and research activities to the public. GCC would not be in Brno without Jana. She's pioneering the uptake of Galaxy in the environment and health research communities through IRENE and PARC to enable fair and reproducible research beyond the laboratory. And with this, I would like to give the word to her. Please welcome Jana Klanova.
Thank you very much, and thank you also for inviting me. It's a pleasure to have you here. Welcome to Brno, welcome to Masaryk University. We will talk today about the human exposome, about exposomics. I will speak about IRENE, which is the new European infrastructure for human exposome research. And of course, a lot of things I will be presenting, it's not really my experimental work, but it's a work of Elliot Price sitting over there and Helge Hecht and their teams. So they combine their knowledge from metabolomics with the bioinformatics to really move the field forward. So few words about our center. We are part of Masaryk University Faculty of Science. Recetox Research Center last year celebrated 40 years of its existence. It started as the environmental center. Over the years, it moved also towards the health, linking environment and health, study environmental impacts, and especially interactions between the society, the environment, and human health. Lately, we also collaborate with the social scientists because, as Helge said, it is also about the interaction of social economic situation with the environment and society and technology. So talking about the human exposome, the whole motivation is that 30 years ago, people believed with the advances in genomics, we will really advance also the precision medicine because we will be able to study associations between the genes and human health. 30 years later, we understand it wasn't that easy, and there are also other factors that we need to consider: the physical, chemical, biological, social, economic, psychological, and others. And this totality of factors we call human exposome. And the field of science that studies all those factors comprehensively over the period of time, it's called exposomics. Of course, you can imagine it's not easy to do it because it's kind of science of everything, but it's still believed that bringing genomics and exposomics together into some kind of integrated framework would hopefully help us with the future advancement of the prevention and precision medicine. And it is a challenge for the whole bioinformatic community. So the history of the human exposome, it's not anything new. It goes back almost 2 decades. The word exposome was first introduced by Chris Wilde back in 2005, and then it was further elaborated by Steve Rappaport. Already under the 7th Framework Program, the European Commission opened the call to develop the concept of human exposome. Then the same things were happening also in the US. The NIH opened the first Hercules Center at Emory University to study human exposome. Then another projects were financed during the Horizon 2020. This time it was 9 different projects, and again, the series of NIH projects on human exposome. Finally, the research infrastructure on human exposome was established in 2021. And you can see that the amount of the papers and reviews and books were growing rapidly. We already have a journal on human exposome. We have several books on human exposome. The question is whether we are there, because we can combine genomics with exposomics only if they are both advancing. So let's talk about some challenges. Going back to the original definition of Wilde, he said that exposome is the non-genetic complement of the genome, the factors that influence the phenotype, which is very broad, very general, and I think it is still a good definition. During the first European call on the human exposome projects, they actually financed 3 different projects, and there was a discussion what is needed to study human exposome, because we need to study those factors over the extended period of time. We definitely need human cohorts. I'm not talking about the clinical studies, but longitudinal population cohorts looking at the populations of different ages. So some of these projects focused on setting up the new population birth cohort. Some were trying to combine existing birth cohorts in Europe. Some were focused more on integration of different omics approaches into the same framework. It was clear since the beginning that we can either study so-called external exposome or internal exposome. So it means looking into the environment or looking into the human body. The first one would mean integrated the information from different monitoring programs, from the Earth observation programs. And it would be very challenging to bring this data together into the framework that would make any sense. The other approach is really measuring the human matrices. But then we are measuring both the exposure markers and effect markers at different omics levels. And of course, we somehow need to combine both approaches, which gave the rise to the development of different variables, personal samples, measuring various factors that are behind the development of the chronic conditions. You can imagine what kind of challenges this means, because if we talk about chemical exposures, we are not talking about 1 or 10 or 100 of chemicals. We are talking about tens or hundreds of thousands of different chemicals, and they all act at the same time. So it's very difficult to study the combined effects of all of those chemicals. At the same time, not all of them are causing the effects, and sometimes the effects are either synergistic or antagonistic. So it's not straightforward. The same challenge is with studying the biochemical or health response, because again, not every biomarker is translated in higher order phenotypic effect. And again, we don't really know which effects are the ones that are responsible for the phenotypic effects that we are seeing. So over the time, the original definition of Chris Wilde was adapted, and it was adapted many times. You can currently find all kinds of definitions. The one that was published in 2014 by Miller and Jones says that exposome is a cumulative measure of environmental influences and associated biological responses. So they actually brought together the factor that is causing the effect with the effect, which is logical because in the biological matrices, you are measuring all kinds of biomarkers. In the other hand, it is very difficult to distinguish between the cause and the effect in such an approach. In another paper published later by Vermeulen, they focused much more on what are the exposome factors, and you can see not all of them, some of them listed on this picture. So it's not only chemicals, but also the physical impact of the environment, the whole ecosystem, socioeconomic conditions, psychological status, and a lot of it. So the attention moved from the chemical exposures to much more general concept of the exposome. And then the understanding was that we could map the external exposome, bringing together all kinds of information in something called exposome maps. And then it is enough to study metabolomics. And some people would perceive the metabolomics as the good proxy for the health. And the attention drifted away from measuring really the chemical compounds, and there were very high hopes for non-target metabolomics that could cover the effect markers together with the exposure markers. Of course, we need to discuss what is the advantage of the targeted mass spec methods and non-target mass spec methods, because environmental chemists typically they work with the targeted methods. They know what they want to measure. They focus on specific chemicals. They have a very specific sample preparation and analytical methods. They can measure their target compounds with a high accuracy and high sensitivity. But of course, they have a limited list of the chemicals they can address. While the non-target method allow us to screen the broad range of the chemicals with a low certainty and a low accuracy, of course. And for those screening methods, we don't really have the QA/QC, the quality control, very well established. And without that, the results that are coming from different laboratories are not really comparable. Metabolomics has additional challenge because there is a very high variability. there is nothing like the stable metabolome for the human body that would be the same over the days or weeks or years. So it means the results are very often irreproducible. They are not very quantitative. They are lacking the power. They are based on the case control studies. They are not easily transferable to the clinical studies. So it means we don't really have a good base that can be easily applied also for the exposomics. And this results in a lot of errors, even in the papers that are published in very high impacted journals, because people are using different sample preparation methods, different analytical procedures. They don't share the libraries, they don't share the standards, they don't really show any QA/QCs. And it's very hard to assess the quality of even the published work. So what it means is that we don't really have the good standards for metabolomics, not even talking about the exposomics. It's very hard to find any information about the raw data, conditions under which the data were collected, about the QA/QC, and data processing, which is most interesting for you, is also less, even less transparent. And into this environment, we are bringing the chemical exposure challenge. With the chemicals, we have much more chemicals than the metabolites. The chemical space is much larger, but also we are talking about the concentrations that are orders of magnitude lower than for the metabolites. So the methods require much higher sensitivity and selectivity in order to really measure the chemical exposures. So in reality, a lot of people are reporting that they were able to identify hundreds of chemicals in the biological matrices. They are either reporting metabolites or the pharmaceuticals or dietary related chemicals, and very few of them are really toxic chemicals that we are interested in if we want to do responsible chemical risk management. And if we talk about the personalized medicine or any prevention, about bringing genomics and exposomics together, we really have to think about the large studies. We need to collaborate with the Centers for Disease cohorts, with the big international cohorts. So we need to bring together the samples, and it would be tens of thousands, maybe millions of samples if we really want to prove that there is a relationship between different exposome factors and human health, which means that we need to develop readily deployable toolbox of the methods for the chemical assessment and stratification, but also we need to bring it together with epigenomics and invest into the geospatial techniques that would link the internal and external exposome. And these toolboxes need to be available to the researchers, because you can't expect that in one institute people would generate the samples from the cohort and have sufficient capacities of the laboratories. So here we are talking about the research infrastructures that need to be developed for that. With that, the European Commission opened the new call for human exposome projects under Horizon 2020. And at this time, it supported 9 different projects. Unfortunately, many of these projects focused more on the health side than on the chemical exposure or exposure side as such. Only the Athlete and Expanse project had a heavy focus on the chemical exposures. And still, they were not really able to develop the experimental framework and a good tool for the assessment of chemical exposure in the future. These projects are coming to the end, and we still don't have this comprehensive toolbox. I said that in parallel, there are many European projects, many US projects. There is also a lot of strategic meetings when we are discussing how to proceed. One of them was the meeting Accelerating Precision Medicine, Precision Environmental Health, demonstrating the value of human exposome, that took place in 2022. And these are the outcomes of the meeting. And you can see they are not very different from what I just said. So it pointed out that we need to establish standardized and robust approaches for biomonitoring. We need to share the data. We need to invest into interoperable data repositories. We need to harmonize the data coming from different human cohorts and merge them together, but also that we need to develop the community of practice. So it means not only discuss, but really start doing that. That created really the need to develop the global community, not to work independently in Europe and in the US, but start collaborating not only between those two continents, but other parts of the world too. So under Horizon Europe, European Commission opened another call for human exposome, but this time they were not looking for the research project, but the coordinated action. So the International Human Exposome Network project is a 3 years meeting project where we are encouraged to identify the people globally who want to advance the field and prepare the roadmap, how we should proceed. So this is about creating the community, but it still doesn't help with technology and the capacity. So at the same time, we really need to invest into the development of the infrastructure and the global landscape of such research infrastructures. And this was actually the motivation why we started developing IRENE, the European Research Infrastructure on Environmental Exposures and Human Exposome. The idea goes back to 2017, when we sent the first proposal to the European Strategic Forum for Research Infrastructures. And since many of you are not from Europe, I should mention that European Strategic Forum for Research Infrastructures is intergovernmental organization. So it's not run by the European Commission, but by the delegations of the member states. And they want to coordinate the development of necessary research infrastructures in the member states to give the chance for the advancement of different scientific fields. So they have working groups on health and environment and physics and social sciences and humanities and such. And we identified that in existing roadmap, there is no link between the environment and health. So environment was mostly focused on environmental monitoring, networks, biodiversity, and climate change, but nothing on chemical pollution. While in the health, it was mostly clinical studies, biobanks, also Elixir as the infrastructure supporting the analysis of the biomedical data. So in 2017, we were able to help to identify the gap in the existing roadmap. And in 2018, this gap identification became the part of the landscape analysis of 2018 roadmap. So that opened the door for the real application for IRENE. So in 2020, we brought together 17 European member states to set up the first proposal for the research infrastructure. You have to understand that the research infrastructures is, the proposals for research infrastructures is not the research proposal that is submitted by the individual researcher or the research community. It is more political thing. So it means the proposal is submitted by one of the member states, and in order to be considered, it needs to be supported by 3 other member states. So it means it really takes time to have a communication with the delegations of the member states. And the whole idea is that this is something that is being co-financed from the European and the national levels. So all the major investments, of course, have to happen at the level of the member states. They need to build laboratories or biobanks or biomonitoring studies or human cohorts. And Europe pays basically for the coordination and the harmonization. European Commission established something called ERIC, European Research Infrastructure Consortium, which is the European legal entity. And when we are talking about research infrastructures, we are really talking about the perspective of 2 or 3 decades. It's not 3 years project. It's really the series of different projects. We are starting with the preparatory phase when the member states have to develop the whole idea, then the implementation phase when the infrastructures are being built, and then the full operation phase when they are providing the services. And service is the key word. Really, the research infrastructure is there to support the excellent science. So it means it needs to be openly accessible to the research community. And it is true for the laboratory capacities, for databases, for biobanks, for the data processing pipelines and such. So some of these existing research infrastructures, for example, BBMRI, which is the European infrastructure bringing together European biobanks, or ERIC, the ECRIN, which is the infrastructure bringing together harmonized clinical studies, they already reached this point. So they were officially set up as ERIC, as the European entity, similar to IARC or EMBL, that is operating at the European level and in line with the European, not the national laws. Of course, you know, for each of them, we need to have the country that is willing and capable of taking the coordination function. And there are always the central services that are looking into the harmonization. So this is a little bit about the background. I said that we brought together 17 countries in the initial phase. Now we are 3 years into the preparatory phase. We already have 20 member states that signed in. And you can see that besides the Europe, we also communicate with the research infrastructures in United Kingdom, also in United States. And we just initiated also the discussion with our partners in Australia, Japan, and other regions. So coming back to the definitions, I said that the original very broad and general definition of wild is still valid, but it is very important to incorporate it into the general omics perspective. So when we are talking about exposomics, we need to see it as a parallel to the genomics. And if we are talking about functional genomics, we also need to talk about functional exposomics, because exposome is interacting with the human body at all of those omics levels. And the most important characteristic is time, because this is all happening over the time scale. We have to think how to set up the framework that allows us to do repeated measurements on different human populations. And then you can tell what kind of infrastructures will be needed to develop this. We will need population studies, biobank, data banks. We need to develop different types of the laboratories. We can leverage the capacities of genomics and proteomics, but we really need to invest into the mass spec-based techniques. So when we are looking at the services that IRIN wants to provide in the future, and some of them are already being provided, we have to think about the chemical profiling, biological profiling. I didn't speak much about toxicology, but in order to assess the relationship between the chemical exposure and the health effect, we really need to talk about mechanistic toxicology. We need to know what are the effects of the individual chemicals. And this is also not straightforward, because typically one chemical has more than one effect, and more than one chemical contribute to the same effect or the mechanism. So the toxicologists over the time developed the concept of adverse outcome pathways, which is pretty much bioinformatic concept. It's like a spider web, bringing together all known information about the chemicals and how each of them contributes to the different biological effects. But again, this is something very demanding because it requires the toxicological community to work in the organized way and to report their information in such the international network. So this we see as the future challenge. And some of you are active in the Elixir community, and you know that recently they established an Elixir toxicological community that started discussing, what kind of support is needed to develop enough computational capacities to support toxicology. In IRIN, we see it as the field of the potential development because it's not only about harmonizing data, but also about, again, harmonizing the laboratory approaches. And then bringing together environmental data, bringing together health data, including the sensitive data, including existing biomonitoring and human cohorts, and also the samples that exist in the biobanks. And again, this is the most interesting for your community, building the data processing pipelines and various types of models that would allow us to bring together social, economic, chemical, biological, health, toxicological, and other types of data. But also what is desperately needed is the central services that will be looking into the quality. And it must be both the quality of the laboratory work, how the data were produced, and the quality of the databases and all the data management process. You may ask, why Masaryk University stepped forward as a coordination of IRIN? It is because the RECETOX Center has been working in this area for 4 decades already, but especially because RECETOX established the national research infrastructure as a part of the Czech national roadmap of research infrastructures already in 2012. So we have more than a decade history of building the research infrastructures with the open access that has several core facilities. We've been running the air and water monitoring networks, not nationally, but internationally. We work as the collaborating center of United Nations Environment Program, supporting implementation of the international conventions protecting human health from the impact of toxic chemicals like Stockholm Convention or Minamata Convention on mercury. We closely work with WHO setting up their global monitoring of human matrices. So we do have the experience with setting up the international programs, but we are also running several population cohorts. Brno is quite unique because there is a history of the birth cohort and other population cohorts going back to 60s, which is very rare in this part of the world. There is a lot of human cohorts in Nordic countries, in Spain, of course, in UK, which is the leader in Europe, but there is hardly anything in Central, Eastern, and Southern part of the Europe. So this is quite unique. We are building on it. We have the birth cohort that was recruited already in 90s, so it is grown up now. We are still recruiting the next generation of the cohort. We have, through our collaboration with University College London, we have the Happy Aging cohort, which was 40,000 people recruited in the Czech Republic, Poland, Lithuania, and Russia. And during the COVID, we also set up the national seroprevalence study on 30,000 people that were followed for 2 years. So it means we have a massive biobank that has hundreds of thousands samples that can support these type of the studies, and it is representative for the region. But also we have the accredited laboratories. Our laboratories were originally set up for the environmental monitoring. So they were targeted and they were focused on chemicals. But in the last year, we are also developing the biological profiling. And once you start generating such a massive data, it is also important to build the data platform. So I'm always joking that we started with EOSC already 20 years before the idea of the EOSC was really introduced at the European level, because we had to develop in-house databases to support those international monitoring programs, but also the data platforms for recruiting our human cohorts, for sending the questionnaires, for keeping the sensitive data. And of course, we had to develop the biobanking system that would keep track of all the aliquots of the samples from our population studies. I said that in the last couple of years, we are also investing into building Recetox bio monitoring or biomarker laboratories, because measuring the chemical exposure and biological effect is using the same technology. So mass spec connected either to liquid chromatography or gas chromatography is the method that is being used by the environmental chemist to measure the exposure to chemical compounds, but it is also being used by metabolomic and proteomic community. So a lot of the tools that were already developed in the metabolomics community can be adapted. And it is interesting to see the interaction between the environmental and metabolomics community, actually, because they are now trying, both of them, quite independently, are trying to develop the field of the functional exposomics. And they are coming with different fields, with different experience and different beliefs. So while the environmental people have a great experience with the QA/QC methods and really heavy focus on the very good accuracy and quantitation, and they are trying to expand their experience to the non-target analysis, metabolomic community is coming with their tools, believing that everything has been already invented. They can just apply it, very often not realizing that we are really talking about the concentrations that are orders of magnitude lower, and that the current technology is not sensitive enough, and current data processing tools are not good enough to be able to cherry pick the peaks that we are interested in. So what is our approach is that we believe that we always need to combine target and non-target approaches. We don't believe that the high resolution mass spec that would be generally applied to the samples that were not pre-processed would give us the answers, at least not now at the current state of the technology. So besides investing into further development of multi-residual methods for chemical analysis, we invested also into the development of the comprehensive suite of the targeted methods for different types of the biomarkers that would describe the health status of the organism, looking into different chronic conditions. And still, we are investing a lot into development of the QA/QC procedures for the high resolution mass spec and the non-target analysis. It has several elements. So one thing is that you can't really check the quality of anything if it is not documented. So step one are standard operational procedures. You need to have well documented how the samples were prepared and how the data were collected. Then the second is spectral libraries, because this is something that is not publicly available. Very often, even the knowledge or the standard operational procedure for building the spectral libraries is unknown. Everyone is publishing some data. They are claiming they are using their in-house annotation libraries. But how can you believe the data if you can't check it, if you don't know how they develop their libraries, what kind of standards they really have, and how reasonable is their annotation? So we've been working with our community, trying to encourage them to share the standards, to share the libraries, to develop something jointly. And it's not going really well, because people are sitting on their own data, believing that it gives them the competitive advantage if they just keep it for themselves. And then we are coming to data processing pipelines, because it's even bigger mess. You can see that there are tons of the different sharewares, the tools that can do something with the mass spec data, but everyone is combining different tools that are available, playing with the data, reporting something that again cannot be really documented. And this is actually the reason why we teamed up with the Galaxy community, because we didn't want to invent the wheel. This was something that was set up originally for the genomic data, and we are offering our capacities to work together on the mass spec data too, because it is very much needed for the advancement of the functional exposomics. And then we are coming to fair reporting, to really the good standards of the metadata, how we want to keep the data. It's not so crucial for the targeted analysis. If you only use mass spec to measure certain masses, then your raw data are not very useful for the future because you already picked all the information from it. But if we use the high resolution mass spec, it's kind of electronic biobanking. So you can always go back and search for more data, but it is only possible if we develop the databases that store the raw data with the minimum set of the metadata standards. So we can go there and look for more, and especially we can bring data from different studies and assess them together. So the first step is really the documentation, and only then we can go to harmonization of between different laboratories. So when building the resources of the standard operational procedures, we focused both on the LC mass spec method and also GC, both liquid and gas chromatography. So it means we are interested in the assessment of both polar and non-polar and volatile chemicals and metabolites. And we are reporting a large set of the parameters. And those are both preparatory, the parameters of how the samples were prepared, and also analytical parameters, which is quite unusual. You can see on the slide a number of the sample prep parameters and analytical parameters that is being reported in our standard operational procedures. And all of those procedures are publicly available. We are using, Zenodo and publishing everything that is available in the house. Again, we are trying to set up the new normal, and we don't have too many followers who would do the same. You would see in the following slides how often our resources are being downloaded, but you don't really see too many people who would do the same and contribute, which is actually one of the reasons why we were setting IRENE and the working groups within IRENE. So altogether, our laboratories already offered 26 different standard operational procedures, and they heavily invested into the harmonization, especially the GC Orbitrap mass spec methods. The important thing is also the libraries. And I can say that we are progressing very fast, because building the libraries means that you need to buy the standards. And they are, of course, very expensive. And we are talking about tens and hundreds of thousands standards that we need to buy, or many of them are not even available to buy. So this is especially why we are trying to encourage our partners to share our standards. And also it is time consuming, because you really need to run those standards. You need to evaluate the data. You need to build the library. So it's a lot of work, and it really makes my heart sink if I think about all those thousands of the researchers who are doing the same work, analyzing the same standards in their laboratories and building their in-house annotated libraries. So we have to change this culture too. Another thing, we just lately had a meeting with the European Chemical Agency, trying to encourage them that they probably have a lot of mass spec data and a lot of standards that are going through the European REACH process, because all the chemical companies that are introducing the new chemicals, they have to report them and they have to provide a sample. So there are the ways how to proceed with the field, but sometimes there are not only obstacles in the minds of the scientists, but also the legislative obstacles that need to be overcome in the future. And then, of course, I'm coming to the Galaxy pipelines. The main reason why we wanted to work with Galaxy is reproducibility. our effort to build the data processing pipelines that are publicly available, that are well documented, that the scientific community can use. Because as I said, there is a lot of people coming from the environmental sciences who don't have really the proper training and understanding and experience, and they would greatly benefit from using the data processing pipelines. Again, like in the case of the laboratory, we are also talking about the pipelines that are built both for liquid chromatography and gas chromatography. We didn't start from scratch because even at the beginning, we didn't want to start building something that other people would avoid. So at the beginning, there was a collaboration with Columbia University and Emory University in the US, because they were the first one investing into building the open access data processing pipelines only for liquid chromatography mass spec. So we really started with their published data processing pipelines. We went through them, fixed the bugs, and then we focused on implementation of these pipelines into the Galaxy. So through the collaboration with the Czech Elixir Society and the people from the Galaxy Society, we had to start building our bioinformatic team who was capable of implementing those pipelines. And then we continued with adding more tools and building also the GC data processing pipelines. Of course, it means building the community, building our own team, building our relationship with other similar institutes, building our relationship with Elixir and Galaxy community. Currently, we released about 20 open access repositories, contributed 59 tools. I don't really need to cite all of it. In the upper right corner, you can see the numbers of the downloads. So this is really something that is being used by the community, and it's very far from being complete. And when people are comparing genomics and exposomic, it's not really good comparison. You know, for the genome, you have 4 building blocks, you have 1 DNA, you can it's much more easier to develop the tools than for this very broad chemical and metabolomic universe. So there is a long way to go, and we really need the collaboration both from your bioinformatics society and also our environmental and health society. We are using different available resources to build the community and proceed with this work. One of them is PARC. PARC is the large European partnership for assessment of chemical risks. It's the project that is supported by 400 million euros for 7 years to really build the European chemical risk assessment. The good thing is that it has one work package that is looking into the infrastructures. So it should map what is available in Europe and beyond, what can be used, and what is the exit strategy? What will happen after 7 years to make sure that all the tools that will be developed within the partnerships will be preserved somewhere. So this is why we are also coordinating this work package on research infrastructures to make sure that anything useful will be preserved and that we can contribute to the harmonization of the processes even within PARC. And there is another work package that is focused on data, fairness of data, open science, again, making sure that all the data produced within PARC are treated well and preserved for the future. So we are using this partnership because many IRIN partners, but are also PARC partners. So we can use some of the activities and tasks of the partnership to contribute to the harmonization process. So within the PARC, there is ongoing inventory of the laboratories, their QA/QC processing, existing data, but we, in the future years, we are also planning to do the assessment of the databases, data processing tools, including the banks of the mass spec data. And we also initiated a discussion about the repositories for the mass spec data. I already explained it's crucially important that we have the good standard for how the mass spec high resolution data is being stored. And actually none of the existing solutions, including the metabolites, is really prepared for this situation because they are typically piling the data coming from people, from plants, from animals. They are not really very well organized. And of course, again, we have no intention to reinvent the wheel. So this is why we got engaged in the communication with EMBL and EBI, that we will work on it together, that within Park and IRENE, we will develop the standards, we will test these databases in our projects, and then we will pass the minimum metadata requirement and the proposed structure of those databases for EBI to keep and provide these services to the research community. Within Park, we were able to set up the collaboration with the Elixir and Galaxy communities in other countries. I should mention at least the Elixir France community that has very well developed the metabolomics community, developing the new Galaxy tools. So this is just one of the examples. But there are still many limitations, and I mentioned most of them already, especially, incompatibility of the existing modules, especially people who are working with different non-FAIR tools and databases and the data processing tools. But there is also not only lack of documentation, but also the lack of the training programs, because exposome became a very popular keyword. Everyone is now organizing the summer schools and boot camps and training courses on the exposome, but again, they are not very harmonized. So we really need to develop the system, and that's actually one of the intentions of IRENE, of the training courses that would train the community towards better harmonization of both laboratory capacities and data processing tools. One of the financial tools that we are also using is the Czech EOS. I mentioned that Recetox, our own infrastructure, is a part of the nationalized landscape of research infrastructures. So we are already being supported by our government by building building the open access services to the international community. But we are also heavily involved in the development of the whole EOS landscape in the Czech Republic. Our ministry had a good idea that the EOS development should be coordinated to a certain point by the existing research infrastructure. So it means we can leverage the experience that we already have from the international projects and partnerships to also introduce the standards at the national level. And the development of the data processing pipeline, mass spec libraries, but also development of the standards for the field of environmental toxicology will be among the national priorities of the Czech EOS. Of course, we are not doing everything ourselves. I said that there is a heavily populated landscape of the European research infrastructures already, and we can benefit from the existing resources. So it means in the health domain, we have BBMRI, which is the network of the biobanks that can provide a lot of samples and data, and we are working closely with them. Once we do have the toolboxes that are applicable in the large population studies, we can also translate them into the clinical studies and work with the infrastructures that are looking into the clinical development, drug design, the models of the disease, phenogenomics, and all those fields. Of course, we are also collaborating with the environmental domain, because this is where a lot of data about the environment is coming from, about the the greenhouse gases or atmospheric particles, about water pollution, food contamination and safety, and all of this. And more we are also collaborating with the social infrastructures. For example, SHARE is the European infrastructure that is operating the European aging cohort in almost all European countries since 20 years. They have a lot of very interesting population, social, economic data, but so far they were not taking any biosamples. So it's a good potential of linking the existing resources with our laboratory and data resources to really advance the field of the prevention and precision medicine. So with that, I would conclude my presentation, hoping that I encouraged all of you to work with us and work with the health and environmental community and help us to proceed with our mission of building the good data processing pipelines to support the functional exposomics. I would like to invite you to two events. This fall in October, we are organizing the joint meeting between IRIN Research Infrastructure and this International Human Exposome Network, where the people will be invited from all the regions of the world. And we are also organizing the joint meeting with the stakeholders. So it means people coming from different research fields, from the societies, from the patient organizations to really discuss what are the main challenges and what would be the best roadmap to develop sufficient capacities for the exposome research. And then the second event is International Conference of Research Infrastructures. This year, this one will be organized in Brisbane. And it's kind of strategic. It's not so much the scientific conference. It's really the strategic conference between the political level and the scientists and the users and the communities to discuss where we should move with the whole field of the research infrastructures. So we are organizing the one session of the conference that will be discussing the human exposome, but we will take the advantage of this conference being in Australia, hoping that it helps us also build the exposome, the network that would include our partners in Australia. So after we closely collaborate with the US, including the NIH institutes and some of their funding bodies, we are hoping that this will also strengthen the collaboration with Australia, New Zealand, and some of the Asian countries too. So with that, I would like to thank the big team that is behind the development of the good tools. I already mentioned Elliot and Helge, but each of them is collaborating, the large team of the chemist and analytical chemist and environmental chemist and the bioinformatician and data scientists. We closely also collaborate with the Recetox research infrastructure, the accredited laboratories, and then the coordinators of the big project. And I cannot forget all the partners of IRIN. Many of them are already contributing to our working groups on the development of mass spec techniques and data processing pipelines. With that, thank you very much for your attention, and I hope that you will have a chance to enjoy Brno. Brno is the city of the history and culture and music. Helge already spoke about Mendel, but this is also the place of Leos Janáček. If any of you is fan of the classical music, not far from here you can find his family house and museum, and there are many other things you can explore. Enjoy the city. Thank you.
Well, thank you, Jana, very, very much for this exciting and great keynote and for opening the conference with that. And with that, we now have a bit of time for questions from the audience. Wait, yes.
Thank you. I mean, that was a very interesting talk. It's great to see that there are systematic efforts to establish and monitor the exposome. And I mean, this is something very new to me. And I was wondering, when you do this kind of surveillance efforts, what does an alarm look like? When would you say, well, we found something new, and like we need to be doing something about this, we will forward this to politicians or whoever can take actions. And then also I wonder, the analysis sounds incredibly complicated. Like I am familiar with genome association studies, but this seems like that squared, really, really hard. So I'm wondering, like, if in any one of these studies, how many problematic compounds do you find, and how many of those are new that weren't already?
Sometimes we say that the journey is the goal.
Yes.
And I think this is
Yes.
this case because we've seen very interesting presentation of the colleague from Imperial College about months ago in one of the exposome workshops organized by EMBL. And he didn't prepare any presentation. He just, took the pen and he drew the curve of kind of this shape. And he said, this is the typical curve that describes the development of the new field, that there is something new. There is a great excitement, a lot of hopes, then nothing is achieved, and then the curve starts declining. And I had in the presentation this, very steep curve going up, and we can't let this happen. So it means we shouldn't be intimidated by the challenge. We just have to be able to have the roadmap and proceed one step after another. So at the beginning, I introduced the whole concept, really science of everything. That's overwhelming. You can't achieve that. But then we decided to focus on the labs. We do have laboratories, we do have methods, we can measure the chemicals. We do have metabolomic people, so there is a lot of instrumental and human capacity to proceed. And we also really need to work with the companies, and we are trying to do that because the technology is not good enough. And the sample prep and everything, it needs some improvement. So that's the challenge for now for us. But there are other groups. There are people that are investing more into this geocode assessment of the geocoded information. So for example, one of our partners is University of Utrecht. They are coordinating one of those exposome projects, and they are heavily focused on external exposome, building the databases, bringing data from different monitoring programs, explaining that part. And then, of course, there are the institutes that are focused more on toxicology. And to your second question, we already have the history of environmental analytical chemistry going back 40, 50 years. So we are capable of measuring hundreds of chemicals. The problem is it is very expensive. So you can't really assess hundreds of chemicals in one method. You have to select several sample preparation methods, several analytical methods if you want to measure polar compounds in the urine, like the phthalates or bisphenols. Or if you want to measure DDT or polychlorinated biphenyls, you typically measure them in the blood and you need to get rid. I said that we want to measure the very low concentration, but if you take blood, it's full of the biological chemicals that with very high concentration, the lipids and cholesterols and proteins. So you basically need to get rid of everything. And here we have a new challenge. We want to measure the biological biomarkers and the chemical biomarkers. So this is why I say that we always need to have a battery of different methods and decide what is the scientific question. There won't be anything, one size fits all approach. And if you ask about what was already achieved, there are tons of the papers in the last 30 years showing the link between chemical exposures and neurodevelopment of children, cardiometabolic diseases. But the problem is they always picked certain group of the chemicals, and those are basically association studies. All epidemiology is about association studies. So we see some type of the relationship between this type of exposure and this type of effect. Are we sure whether there is really a mechanism for it? So now we are saying, okay, let's study more comprehensively variety of the factors, variety of the effects, but also find those links.
Okay, there was another question. Yes.
Hi, Florian here from the German Cancer Research Center. Thank you very much for the talk. It was a great overview. I have two broader questions. The first thing is, I mean, we deal in omics, genomics already, and still with a huge gender and ethnicity bias. So how do you deal with that in Irene/Park? That's the first question. Second question is, are you also considering to refurbish basically existing data? You mentioned already there is already a lot of data, but specifically I'm talking about now rare diseases and pediatric diseases. So where we don't have so much data, so we already have not enough data, and the data we have is not really standardized, not harmonized. So do you consider this data as well to maybe bring it to a standard? Maybe it's not perfect, but then we have still some data to analyze, basically.
But I think step one is to set the standards because they are missing. So I think this is now most urgent to really decide what are the standards for the sample prep, for the data quality, for the management, for the data processing pipelines, and then offering them to the community. So we don't lose more time and we start collecting data comprehensively. It's not only about having a good standard, how we measure things, but very often you can just add additional method into the existing study. This is why we are trying to reach out to the medical community, because they have a lot of clinical studies. For us, you have much better chance to identify some effect of certain chemical in the clinical study where the chronic condition already has developed, rather than looking into the healthy population and waiting for 50 years before, somebody develops really, the sign of cancer or neurodegenerative disease or something. So we are really now focusing on setting the standards, identifying the key partners, convince them to provide enough capacities for the storage. So once we start collecting the new data in the right way, we can go back and see how much can be adopted. Of course, we can't waste of the existing data, but you mentioned rare diseases. That's number one, why we really need to develop those toolboxes, because they are rare. We don't have enough data. And if we waste the opportunity and are not able to bring together data from different countries or continents, we will find nothing.
Yeah, and the first question to the gender and ethnicity bias, are you considering that?
That's something, it's more a question for epidemiologists. So they, again, it's associations. They are looking at these parameters in their data, not really being able to explain them, but they consider them. In the measurement, we don't really have too many international studies. So if there are any population studies, it's different for the clinical studies. They have much broader data, but so far we didn't collaborate enough with them. With the population studies, typically population cohorts are national. If there are any efforts in the Europe, it's still quite homogeneous population. This is what we did in some previous joint programs or European projects when they pulled together 7 cohorts or 15 biomonitoring studies, but the European population is still homogeneous. And especially for the longitudinal cohorts, the requirement very often is that the person recruited needed to live in the country for a couple of years. So you are also kind of excluding the newcomers and immigrants and students and commuters. So we first need to set up these international collaborations. And this is also one of the reasons of this International Human Exposome Network and our IRENE effort to work closely with people in Australia, in US, in Japan, in Asia, because then we can have an access to heterogeneous populations and compare data.
Okay, thank you.
Mm-hmm.
Okay, we have time for one more question. Yes.
Thanks a lot. I was especially blown away by this interdisciplinary aspect of your talk, right? Or of this entire field. And I'm wondering, so we have here now experts from climate science, from earth science, from imaging data analyzers. So do you actually have this data already so that we can link now genomic data to chemical data to climate data? Or is that something that we need to wait for the next years to bring that together? So I'm especially envisioning at some point really an integrated data analysis where we can map climate data to exposome data and maybe even then to genomic data at some point. How far is that in the future, or do we have a few data sets where we actually demonstrate that already?
It is happening. This is what people are already attempting with the very questionable results. Of course, you can try it on the smaller data sets that are available. You can't really have very high hopes, but technologically, you can start working on it. For example, we just had a meeting last week at a local level between the climatologists, people that are dealing with the biodiversity, and the social geographs. Because especially for this region, we really have a lot of data. We have 40 years of our own environmental monitoring. We have closely collaborated with the city of Brno that has also the information about the social stratification of the region, about the traffic and the pollen events and all kinds of data that were ever collected from the South Moravian region. So they made this data available for us. But then the social geographs, they can link it to the census data, to all the information about the social economic status. And then when we had a discussion with people who are climatologists, they have a lot of data on microclimate within the cities. So they can go almost to the level of the street and the square and have the information about the temperature pressures and heat waves and these kind of things. And it is interesting that also people dealing with the biodiversity have very detailed information about the species within the city and the region of the city. Because when we are talking about the exposome, one of the things that is being discussed is the accessibility of the green places and blue places. So it means people feel generally better if they have an access to the park or walkable distance to the forest or the lake or something. And this is again a lot of information that people, the biologists studying the biodiversity have. There was even one call from the Horizon last year that was asking the same question: is there any difference between the biodiversity, natural biodiversity, and the well-being? So there are first attempts, suggestions that we should look at the concept. A lot of people blindly believe in artificial intelligence. They hope that artificial intelligence takes care of everything, but it is basically garbage in, garbage out. So unless we have a good control of the quality of the data and the way how the data were collected, we can't believe in the results, but it shouldn't prevent us from developing the tools. So one thing is looking into the quality. The second, we can go ahead and use existing data and test some new concepts.
All right, good morning, everybody. So before we get into the talks, I'm going to spend a couple of minutes giving you an overview of what we have coming up and introduce sort of the scientific program for GCC 2024. So with that, I'd also like to welcome you to Brno. I've spent 2 days here and have been absolutely wonderful. And I hope you have a great time with everybody that's here in this space. So for those of you that don't know me, I come from Johns Hopkins. My name's Enes, and I'm the chair of the scientific committee for GCC 2024. I've interacted with many of you, and if you don't know me, feel free to come up. If I see you or as I see you, I'll come up as well. And one of the things that that I'm responsible for is this scientific program. So if you have any comments for this was good, this was not good, this is how we could make it better next year, please come and talk to me at any point throughout the week. So we're all here. We've traveled from various corners of the world spent a couple of days in why did we come? Why did we take the week out of our lives to spend here? And so sometimes, and I know many of you personally, we like to travel, and so coming to a destination that's new to many and away from home, if nothing else, is often exciting, right? And Brno, as we've heard this morning already, has some historic significance, has some cultural opportunities for us to explore, and that in itself is a great reason to come to GCC. As you'll hear more about and as you'll experience on Wednesday, we embed a half a day tour of sort of the region and the city to give you some cultural experience in addition to the scientific kind of growth opportunities. We also travel for food, right? I had the pork knee the other night, and it was absolutely wonderful. If you don't eat meat, there are other delicious things on the menus usually. So that too is a reason why we travel. Last year when GCC was introduced, we also traveled for beer. That was the Czech Republic apparently drinks the most or beer per capita of anywhere else in the world. So, of course, we have to sample what the local culture has. But we also come for science, right? I mean, that's really, in many ways, the reason we come to a conference, and GCC in particular. We invest a large portion of our year to develop cool new things, and we want to share it with our colleagues. We want to learn from our colleagues and see what are we going to spend the next year on. And we also come for the community. Right, we interact with each other. Like I said, I know many of you, most of you personally. I hope to meet most of you, the rest of you, this week, and continue that, develop that friendship over the coming years. And so, but all this sort of general concepts aside, I want to go over to some specifics of the scientific program that we actually have coming up over the next 3 days. So we have sort of 3 themes that have been contributed to by the people in this room, meaning they haven't been invited. So we have talks, posters, and BOFs, or birds of a feather, as high-level sessions. We have 2-hour sessions every day. And the thing that we're doing a bit differently this year than previous couple of years, and I think maybe the first time since Freiburg in 2019, is we have today's track and the Thursday afternoon's track is single tracks, but the 2 tracks tomorrow and Thursday again morning are double track. So there's a more sort of analysis focused track, and there's a more what we call enablement track, meaning technology developments that help or encourage analysis. And something to keep in mind is all talks are 10 minutes. So we did away this year with the short and the long format. And we're going to experiment and see. So again, one of those things about, what do you think about this format? Please share that at the end of the event. The schedule is on the website. That's the most sort of up-to-date schedule. So if you ever wonder what's coming up or what track I should go to, just check the website. As far as some stats goes, so we got 54 talks, and it was authored by 339 people. It's comparable to what we had last year. But we got 10 hours of presentation. That's due to this double track that we introduced. So it's 2 hours more than we had last year, and an opportunity to, again, learn about a lot of the work that people have spent over the past year. We also have a lot of posters. We have one more poster than talks in terms of the numbers, unlike last, so this is, I think, the first year we actually have more posters than talks. There's 33 that are posters only, and 22 that are posters from talks. So, and on top of that, we did a 2-hour poster session this year, as opposed to the 1. There's coffee along the posters, so you kind of mill around and chat with people. There's no pressure for squeezing 30 or so interactions in just 1 hour. And then the other thing is the way the schedule is laid out. Most talks that have a talk in the morning have a poster in the afternoon for those that have elected to have a poster. The Thursday talks, unfortunately, we couldn't make that happen, so they're on deck for this afternoon. We also have BOFs, so birds of a feather for those of you that are new to GCC or BOFs. This is community organized events that are just organized as one wishes. So you can put forward an idea for a BOF, show up, you can have some talks, you can just have a discussion, brainstorming. They're a great opportunity to meet people that are interested in the same space, Great opportunity to share some early ideas, get some feedback on those. And so we have 13 of those this year on a variety of topics. So look over the list and go to whichever one or jump between them as you seem appropriate, as you seem desirable. And then with that, I want to thank the scientific committee. So the names don't match the pictures. That's intentional. I realize that. And so I did that so that, for those of you that know the people, you take a moment and try to match the name to the face, because otherwise you just kind of forget the names that are and the pictures that are on the screen. If you don't know the names, you can see the people in this room and go introduce yourself to them. And so these 6 individuals have done 175 reviews of the abstracts that people submitted. And they made sure that this content is scientifically sound and interesting, so that we, the 150 people or so that are here in the room, have something interesting and engaging to participate in throughout the week. But now with the corrected names, I also want to say that this group of people is not some group of senior prestige scientists that sit high on a chair and evaluate what people have done. They really have the privilege, and more like this little boy, the children, the scientists that have the privilege of being the first ones to learn about the work that you all have been doing over the past year, right? It's this excitement, it's the opportunity to learn firsthand about a lot of work that has gone in the galaxy world. And so I want to take this as an opportunity to recruit some of you for next year's scientific program. If you are interested in being one of those people that does the early readings and reviews of the content, please check out there's a QR code and the link at the bottom that you can submit an expression of interest form for joining the committee. And so in addition to the posters, the BOFs, and the talks that I mentioned, we have a few elements of the scientific program that or people that have a lot to share with us on a larger scale. And so we've already heard the keynote by Jana this morning. We have 2 more keynotes coming up, one on Wednesday, one on Thursday, and then tomorrow we have the Galaxy Community Update talk by the PIs of the project. And so again, show up early in the morning to see what these individuals have to say about the future of this field, or many fields, as it turns out from this morning's keynote. And then for the first time ever, we'll have a panel. So we have a group of people on Wednesday mid-morning that'll talk about 3 high-level topics, I guess, about empowering users, increasing the global presence of the project, and the project growth and sustainability. So this is a bit of them reflecting on these topics, and a bit of an opportunity for you all to ask questions about the project and learn what these people think about it. And then whether we're in the sort of later in the career stages as a senior scientist or early students, those publications do matter. And so you've gone through the effort of submitting an abstract, preparing some slides, possibly designing a poster. And so I would strongly urge you to publish this online in a tractable and in a long-standing format, so with a persistent DOI. So Galaxy has a partnership with the F1000 Research. It's a publisher that allows us to upload our documents, get a DOI, and a way to publish site that we've done. We're also doing something new this year, thanks to Wendy, who suggested it, and thanks to Marissa for leading this effort, which is a student-led meeting report paper, meaning that it'll be published in the F1000 journal as a sort of a summary of the activities that have gone on throughout this week. There's a BOF on Thursday afternoon that will sort of start this process and get all the students that are interested in contributing to the paper in the same room and learn some, here's some lessons, I guess, from others that are going to attend the space. Then in addition to all the talks and sort of short format learning opportunity, there's a lot of training at GCC, right? There are 32 training sessions spread across 3 different days, totaling 9 hours, and we have 34 people presenting these topics. So if you have questions that aren't necessarily answered in those trainings, or extend the training, those are the people that you want to ask. And with that, I want to thank Tyler, who, in my opinion, got a new title, the cat herder, for the first time. He had to negotiate, I guess, with these 34 individuals to get all the content and schedule on time. So thank you, Tyler. And with that, I'll wrap up. I want to be a little longer, but we have 10 talks this morning that are listed here. And so with that, I'll invite Tyler and Alex as our first speakers for the BioDigs and GDSCN project.
Hi, everybody. I am Alex Ostrovsky, and this is Tyler Collins. We're really excited to talk to everybody about BioDigs, which is a new project Galaxy is getting involved in. Galaxy has historically grown quite a bit with our collaborations with outside organizations as well as larger scale projects. We've been working with the VGP for a few years now, which has really enhanced our large scale genome assembly capabilities. We've been working with Anvil for several years, which has led to a lot of enhancements in our cloud computation capacity. And our big projects on COVID have really enhanced our workflow capabilities. We're really excited today to talk about BioDigs, which is a project of GDSCN. GDSCN is the Genomic Data Science Community Network, and it works to promote research opportunities for underserved universities and students who would not necessarily have the opportunity to work in large-scale scientific efforts. Currently, it is mainly in the United States, but we're going to be talking at our BOF later, hopefully, about how to expand that and what that would look like. A little bit about what we're doing right now. We're looking at genomic diversity in soil. Soil is the most biodiverse substance in on the planet. 1 gram of it contains billions of cells representing tens of thousands of different species. Soil is home to 59% of life. It is the singularly most biodiverse habitat on Earth. That said, what is BioDigs? We're asking a few questions about what we can learn from the soil, microbiology and metagenomics questions. What is in the soil? How does genomic composition change over time and location? Genomics and bioinformatics. How do we ask those questions? What is the best method for getting those answers? Agriculture and environment. How does this affect what we're growing, where we're growing things, people living nearby, and public health? Soil is important. Kids play in soil. You eat food that comes from the soil. Probably want it to be healthy for everybody. It's also really good because it works with the Galaxy ethos to really involve the community at every level. For students, we're going to be providing Galaxy trainings and support. Computational resources, obviously, big thing for Galaxy, regardless of where your school is and what resources you normally have access to. And the possibility of publications, which, as Ennis mentioned, are important for getting your career off the ground. For professors, we're going to be having pre-designed lesson plans that are going to be published on the GTN with specific deliverables, always a good thing for teachers. Access to Galaxy community for help, and the ability to avoid student dependency problems across different operating systems. It's going to be much more easy for professors to teach with Galaxy than having to deal with everybody working on different OSs. And for Galaxy, new tools, IWC workflows. It gives a new starting point for trainings. Historically, we've done a lot of RNA stuff, and this is just going to be another place to start. And new Galaxy publications to get our name further out there and have more people using us. Now, I've talked a lot about what this is going to be doing for the Galaxy community, but I have yet to really mention what we're actually doing. And for that, I'm going to hand it off to Tyler to tell you a bit more about what BioDigs actually is doing.
All right. Thank you, Alex. So BioDigs is really focusing on 3 scientific goals. We're trying to identify genomic diversity across the soil samples, ideally identifying new species of both bacteria and archaea. Currently, most of the species present in the soil cannot actually be cultured in labs. So there's a huge lack of information about what's actually present there. Additionally, we're trying to find environmental associations, especially looking for metabolomic pathway analyses, trying to identify what's actually occurring in these soil samples across the United States. And lastly, Key part is trying to identify how any of this relates back to human health and trying to find any human health connections. So to that end, an initial pilot study has been conducted already. We had partnered with several colleges and universities in Baltimore as well as Montgomery County, which is a county right outside of Washington, D.C. From these, we sampled 48 total sites, 24 in Montgomery County and 24 in Baltimore with a couple replicates. If you look here on the right, you'll see some of the sites, and these were all partnered with students. And essentially, these soil samples were taken. They were sent to both the University of Delaware as well as the Johns Hopkins Sequencing Corps. And from that, we did both Illumina sequencing as well as some PacBio HiFi sequencing and some soil metadata analysis. So looking at differences in lead concentrations, heavy metals, biological materials, pH, and several other more common soil samples. So from that, we had some very early results to share with you today. This is genetic diversity generated from a tool called SILF. SILF, if you've ever heard of Kraken or worked with Kraken before, is very similar. It uses kind of a k-mer-based approach where you compare the k-mers from your soil sample against a known genetic database. And from that, you try and identify species. So in this, we have our species, I guess, separated by orders and clades, not species. If we try to do individual species, this became illegible very quickly. But of particular note from a first look is we have pretty high different concentrations of orders of bacteria and archaea across these different sites. A particular note from what I was looking through and analyzing some of the sites and site data was the high presence of acidophorales, which is known to to exist in high prevalence areas of iron and sulfur, and is usually found in mining runoff areas and areas that had heavy mining. And these were found in a nice little park in Montgomery County, which seemed quite odd initially. Additionally, here are some of the early heavy metal data still showing that order clade analysis. I won't go into a lot of the genetics here, but of particular note, so of these 48 sites, we were measuring heavy metal concentrations of lead and arsenic. In almost all of them, they were above the legal limit of lead in milligrams per kilogram concentration in Vermont. That changes a little based on the US states, but it got shockingly high in quite a few areas. Additionally, the US does not have a limit on how much arsenic can be present in soil, but the EU does. So we added that fun little line there, and you can see there was several sites in the US that were also above the legal limit of arsenic. So even in these pretty well managed public park areas, university campuses, very common areas where you'd see where people are playing and active in communities, there's probably a lot of heavy metal contamination, and we're very interested in trying to look through and see if there's any associative bacterial archaeal populations that may have negative impacts to human health in addition to the negative impacts of just the high metals. All right, so to go a little more broad again about how we're working with our other partners. So into Galaxy from this, we've been adding some new metagenomic tool suites. I briefly mentioned SILF, but SILF is a new one that's coming in. We've also been pushing to add in the new taxonomic databases. A lot of the soil metagenomic databases are constantly being updated and expanded, so we're trying to include those within. Additionally, we're developing several new workflows to try and push through into Galaxy ultimately, some focusing on an assembly-based approach, as well as some of this regular taxonomic identification, and hoping to include improved long-read technologies to the platform as well. And to that end, we've also been partnering with some of our students. So these are both Catherine and Nia. They've also been working on the BioDigs project, and they've been developing their own workflows in Galaxy, as have most of the other people participating. So it's just sharing some of the other analysis. In this case, it was trying to identify different BCG abundances ultimately, going from raw Illumina data assembly and going through. So we've had several other schools and a lot of the students going through Galaxy and also developing their own workflows, trying to use some of this early data. And just to save time for questions, I'll go into a brief summary. So the main goal of this project really is to increase equity in science. It's to try and bring in the power of genomic data to people who historically wouldn't be able to use the computation for it, as well as to try and actually do real statistically meaning science and try and investigate soil, which is something that people interact with every day. It's what your food grows in. You're touching it all the time. And it's kind of this big gray area where there's been very little research historically. So trying to combine both increasing equity as well as identify and research something that's a little unknown and very important. And from that, I'd also, as Benna said, like to recommend you guys come to our BOF. It's this evening. So if you want to stop by, we can talk about both what we've been doing and how we're hoping to expand the project and get more people involved. And then from that, I'd like to give a lot of thank yous and acknowledgements to all the students from all the different universities who've been involved, as well as some of the GDSEN team, as well as organizers from Johns Hopkins, Cold Spring Harbor, PacBio, and several other companies that have been assisting with us. And from that, I think we can take any questions.
Thank you, Alex and Tyler, for this great presentation. I have a question. Do you collect any metadata, or what kind of metadata do you collect for this soil sampling? Because there is, for example, a huge link of soil and mycorrhiza in the soil links to the connection of trees and the communication of trees to each other. So it would be really interesting to have like metadata which is connected to the forest itself. So what kind of metadata are you collecting right now?
I don't believe we are currently taking data about mycorrhizal connections at the moment, but that is a fantastic idea, and I love mycology, so that's something I'm going to probably follow up on.
I can say I know we were taking in standard soil description quality. So basically sandiness, what kind of the loam is. We're pulling in pH, latitude and longitudinal data. We're pulling in metadata if it's managed or unmanaged land. And there was a couple more that I'm sure I am forgetting as of the moment. But part of the goal of some of that initial pilot study was to try and expand some of the workflows and processes to help deliver to people. And one of that is also improving our metadata collection and trying to ensure it. We've also been maintaining photos of all sites. So every time there's a soil collection, we have photos of the terrain, the surrounding area, and exactly what was there.
Okay, thank you.
Hi. Within your EMPL flagship project, doing the track expedition, where essentially it's like European coastlines, they're taking water and soils like that, doing somewhat comparable work with chemical profiling, etc. I just wondered if you'd been in contact, because it would be good to then kind of harmonize.
So currently we're funded basically exclusively for the United States. However, that is part of what we want to talk about at this BOF, is specifically what we can do to make this a more international effort, because we value our international community.
Just because I can pass on the connections to the people lead.
No, that'd be very helpful to be able to talk to another national group that's working on the same kind of project.
Right.
Currently, I do not believe we've had any communication with them.
Right. It's large. So
Love to hear it.
Cool. Great. Thanks.
Okay, good morning. My name is Miroslav Ruda. I'm from CESNET, but what I will be speaking about is just few activities we selected to present it here. And I'm mostly teaser for you to make you convinced that you should go to our BOFs or to our presentations and so on. I will be speaking about few topics which are enlisted here, and to save time, I will just skip to the first one. Just one slide to introduce us, why we are here and why infra, what is it? Infra is national infrastructure in Czech Republic. We cover almost everything you can imagine as a infrastructure, covering from networks, grids, clouds, HPC, Euro HPC, everything pushed by government together to one infrastructure. Even we are still three institutions. CESNET is something you probably know as an NREN, National Research Network Provider, but we cover also grids, clouds, and so on. Then there is IT for Innovations, our Euro HPC Center, and there is specific site, CERITES-C, which is from Masaryk University, from here, from Brno, which covers specific cloud use cases. I will be not speaking too much here, but you can ask me if you have some questions. Most of the infrastructure which is somehow integrated with Galaxy is what grid, clouds, and all this stuff means, and this is covered in Metacentrum. here you have some numbers. I'm not going to read it, but what we really provide is great cloud clusters, Kubernetes clusters, OpenStack infrastructure, and so on. The same story in the pictures. We are spread across Czech Republic. So you are here in Brno, where is major one big center from Metacentrum. Then we have big installations in Pilsen, Prague, and some other across the Czech Republic. We also integrate resources from Elixir. Those are in this color. And we have also nice collaboration with data storage part, which I will not cover in my presentation. But again, we have repositories across Czech Republic. So what we really do with Galaxy is that we support 3 big or main installations of Galaxy. We have chosen use Galaxy CZ as a main gateway to our infrastructure. So we don't provide yet another just for Czech Republic. We really provide just use Galaxy.cz, and everyone who has life science AI, which is generically publicly available, has access, and Czech people has a special privilege if they are recognized in Czech infra AI, then we can recognize their projects and give them bigger quota or something. But you as a general users or Czech users are going to the same server, to the same infrastructure. And the Galaxy can submit to any clusters we have. So again, there are quota limitations and so on, but yeah, if someone agrees with us on some project, he can get access to any resources we provide. And then we work with few groups, and we are building the specific installations. You have heard about the Recetox today morning, so I'm not going to cover this last installation, but we have also flagship project in Elixir CZ, which is Repeat Explorer, which has some specific users. There are just 2 or 3 tools which they provide with some necessary tools around, but still they have more than 1,000 people running their computations. We are part of a Euroscience Gateway project. You have heard about this project last year, and I'm pretty sure that there will be some presentation this year too, so I'm not going to speak about the project itself. We have just 2 tasks which CESNET is responsible in the projects, and both tasks are represented here also as BOFs or some other presentations. One is that we developed test interface for the Pulsar. Pulsar is something which sits between the Galaxy and the clusters and can represent different type of clusters and so on. And the test is task execution service standardized API, which was defined and developed by GA4GH. It's Genomics Health, and this is general something, I don't know, see. So they defined the standard for task execution, which is something which is really just one computation, should be bundled somewhere in the container and run on one side, and the workload execution service, which is somehow related to the Galaxy or very similar to what Galaxy is doing. The complete workflow can submit different tasks to different sites even, and so on. So we developed implementation of test interface for the Pulsar. So, and we will show you some results on one of our demos. And then we just started on something which is called visualization of Galaxy and Pulsar networks. In the projects, we have many Galaxy servers across Europe. We know that there are others around the world, so we need some online visualization, history visualization, how many jobs are currently running, and so on, so on. Again, we have both, so I'm not going to the details. And the last part, which I should at least cover for a few minutes, is another big development project in Czech Republic, which is running under umbrella of EOSC projects in Europe and Czech Republic. And this is what we call national data infrastructure, which is something you can imagine as really the capacity for research data created in Czech Republic. This should be stored in repositories, thematic repositories, which are managed by projects, by scientific groups. We, as the infrastructure, are covering basic design, coordination, and basic services, which are listed here. The whole project is just started, almost just started, and the result should be, and it depends how you count the numbers, but the result should be something like 50 petabytes of real user capacity, which can be used by users. How is it created? And just on the last slide, and this is also description how Galaxy is related and integrated. The Galaxy was selected as one of selected workflow systems which will be integrated with the repository and will really serve as a gateway between the computation and storage part. And if we will cover basic use cases, you can imagine user is doing some computation. When the computation is finished, data or data set can be created in some repository, thematic repository, which is corresponding with the computation. The user gets back permanent ID, which can be then used, and the both workflow and data can be stored in the repository. Metadata should be created. I should say that the repository for us means both data and the metadata, which are describing how data was created, what's the meaning of data, what is access policy for data, and so on. The Galaxy should allow us to cover things like reproducibility and semi-automatic metadata data preparation and so on. Once you have a data and workloads in the repository, and someone different can download data using the PIT and so on, and could do computations, their own computations on the data. Someone else can reuse your workflow and run it on his own data, and so on. So the technical part which we should cover is integration both with repositories. We will have at least 3, but 2 are probably known for Galaxy community, and the background will be S3 storage. So we will also use and leverage S3 integration with the Galaxy, which is already there. Our task will be just integrated, and we'll develop some necessary improvements, and that's it. And for last slide, you have some links. And I should also mention that the group we are running Galaxy are from here, from Brno. So if you are looking for some suggestions where to go to the bar or to some entertainment, we are your guys.
Thank you, Miroslav. Do we have any questions from the audience?
Hi, thanks for the talk. My question is in the National Data Infrastructure project that you are involved in, I presume that you're dealing with data from many different research areas. And if so, which areas?
Any area which is really in Czech Republic. We have 5 working groups which covers really anything: life science, physics, every social sciences. And the 5th is I don't remember the 5th one, but really any science in Czech Republic should be somehow connected with us. We have 10 partners in the project which are representing different areas. And then anyone in Czech Republic or any group in Czech Republic can create his own repository with its own metadata standards and descriptions and so on.
And the workflow management system is Galaxy.
We will support 3 this workflow system. One is Galaxy, one is Lexis, which comes mostly from the Euro HPC world, and we will for sure support also Jupyter Lab, Hub, and all this stuff.
Thank you.
Good morning, everyone. My name is Junhao. I'm a software engineer at Murphy Cancer Center. And basically, I'm going to introduce the progress of Galaxy ML tools. Galaxy ML tool is a tool that aim to provide the simple, powerful, and robust machine learning tools for Galaxy and help data scientists to doing the data analysis using machine learning algorithms or deep learning algorithms. And first of all, what is machine learning? Machine learning is a set of algorithms that can learn from data, and then we can, after training, we can use the model to make predictions on unseen data. And also, supervised machine learning is a category of machine learning and use the label data to do the training and then also make the predictions on the unseen data. And we list a few examples of supervised machine learning in biomedical data analysis. The first one is we can use machine learning to predict the therapy response in cancer from RNA sequence. This also the example application we will share today. And the second example is that we can use machine learning algorithms to do the predictions for age from RNA sequence. And the last example list here is we can use machine learning or deep learning algorithms to do the predictions for IHC status from H&E images. And Galaxy ML is a tool set which has been done in the past. As you can see here in the panel A, we can first step we can define learners. It can be traditional machine learning learners or deep learning learners. And the second step, we can give them an input, and we can train and evaluate the model, the learner. After training, we can do a lot of like plots or visualizations to understand the performance of the model. And in the panel B, as you can see, that is a method for example. You can define something like random forest to training on your data, something like that. And the panel C is a workflow to do a hyperparameter search for a model. And we are always thinking how we can make the Galaxy ML tool better. We conclude a few pain points here. The first pain point is how we can build and reuse the complex machine learning models or deep learning models, especially for the deep learning models. Sometimes it could be many layers, like less than 15, something like that. And sometimes it would be very hard to build a model. And the second pain point is how to define the different parameter, especially when we do the hyperparameter search. And also the how to make the better plots to understand the machine learning black box. And also how to use the pre-existence or pre-trained models, like how to use the pre-trained ResNet 15 on our own data, something like that. And Logwing is a framework we think that could be a great solution for these pain points. As you can see here, we just need a very simple config file containing the input features and output features, and we can use the command lines of few lines of Python codes to run the very complicated vision transformers on your own data, and then we can have trained models on unseen data. And PyCare is another framework or packages with things we do believe is great solutions for traditional machine learning algorithms. As you can see here, we just need a few lines of code, and then we can get the results of different machine learning algorithms on the same data set. And about the Galaxy and Logwings about the Logwings and PyCare in Galaxies, we made the videos to display the functionalities. Thank you. What is to have a video. Yeah. Galaxy. Hello everyone, I'm very happy to introduce Galaxy Logwings and Galaxy PyCare. And we describe these two tool sets to Cancer server. And we create the Galaxy ML tools. We can see different Logwing tools here and also the PyCare tool. For Logwing, we have Logwing config generator to auto-generate a config file from a dataset for training. And having the config file, we can use Logwing trained Logwing hyperparameter search and also Logwing experiments to train the model. And having the model, we can use Logwing predict or Logwing evaluate to predict some samples or evaluate the models on testing sets. And we can also do the visualization here. And for PyCare, we have the functionality to compare different machine learning algorithms on the same dataset to see which machine learning algorithm is better for this for the specific dataset. And let's jump into the example application here. We are today we are going to use the RNA sequence to predict therapy response in cancer. And basically, this is a binary classification test, and we have 7,000 input features. And and having the dataset, we can use Logwing config generator to generate a config file for training a dataset, training a model. And first we select the datasets, and second step is to select the output feature, which is also called target feature. And after running, we can get the results like this. These are pretty huge config file because we have 7,000 input features. And then having the feature config file, we can use the Logwing experiments to train the model. First step, select the config file, and the next steps giving the input dataset to the to Logwing, and we can run the jobs. After running, we basically will get three results. The first result is the prediction CSV, and the second result is the model. You can download the model here, and the last result is the HTML report. From the reports, we can see the different plots and the performance. And that's basically for Galaxy Logwing. And right now, let's jump into the PyCare. For PyCare right now, let's select the input data set, and the next step also select the target columns, and we can use for classification or restorations, right now classifications. If we want to change the default parameters for training, we can definitely do that here. And after running, we will also get three results. The first result is the model, and the second result is the dashboard.html from Explainer dashboard. And you can see here, we can see the feature importance or different plots and feature dependencies. But we also have another HTML for the parameters of the best model, and also for the results of comparing different machine learning algorithms on this data set. And we have more plots here. And that's basically the introduction for Galaxy PyCare and Galaxy Logwings. I hope you enjoyed this video. Thank you. Sorry, just I speak out the videos because of the time limitations. I can do a live demo this afternoon to you guys if you're interested in. And for the future words, we will continue integrate the expander dashboard into our toolbox and to provide the better visualizations, and also help data scientists to understand the things behind the machine learning black box. And we will also develop more PyCare features into our toolbox. And we will also implement training material for Galaxy Logwings and Galaxy PyCare to create more histories or the documentation about how to use the machine learning tools. And I can take one question, I guess.
We got one super quick question. Otherwise, come to the poster this afternoon and talk to you and how. I will go with the poster. All right. Thank you again. Oh, sorry. There's an online question. Can't be that. Okay. There's an this seems generic. It could be used in any field, not just bioinformatics. You. Yeah.
I totally missed the question. What was the question?
This seems generic. It's not just tied to bioinformatics. How could it be used in another field?
You mean it's probably it's not. I would say it's this basically the tool set is for any kind of like images or CSV data or something like that. So I think it could be used for any kind of fields. Does answer your question?
Well, it's the online question. So if you don't mind answering in more detail or starting a conversation online on Slack, that
Yeah,
it would be appreciated by the person asking. So again, thank you.
Hello, everyone. I am delighted to introduce our first attempt at doing community lightning talks, at least as long as I've been in Galaxy. So this is on behalf of the Galaxy Community Board, which if you don't know what it is, you'll find out bright and early tomorrow morning. So we're going to start with my favorite community of practice, the single cell and spatial omics community of practice, which is a mouthful. So we've rebranded as Team Spock. So we are a lovely group of people, and big shout out, I have 3 students with me who are part of this community today, Marisa, Morgan, and Julia. So please do talk to them and show them how wonderful this community is this week. So why do we exist? Well, we originated because we've realized that 2 separate groups had wrapped the same tools at the same time. And we'd really like to avoid that in the future because there's quite a lot of tools we need in the single cell space. So our main goal is to talk to each other so that we know what we're doing so we don't do the same thing twice. Our second goal, if we can manage it, is to also try and identify and address user needs. We work really hard to get users and developers to talk to each other. So we get both the users giving feedback, we get developers helping the users then feeding back on this process and trying to really, do the full loop within creating software that helps people. What have we been up to? These are just some cool community features that we've been working on to address some needs. So a big thing for us was trying to make communication easier. So a big shout out to Nate and Helena, who created this Slack matrix bridge. Matrix is where Galaxy lives, Slack is where training lives. So trying to get users to stick around after a course and keep talking with us is now enabled with this, which is awesome. We've also piloted a tool request sheet, which we're going to try and again get users to engage more with. We've made some nice FAQs to point people who get to our training to use our subdomain page, which is easier for beginners. And yet again, we managed to, before I came along, have 2 different subdomain pages. So we did unify those so it's a clearer user trajectory. How do we function? We really try and do best practices in meetings, which I think a lot of the groups in this organization try and do. So we have monthly meetings. We roll across all the time zones to make sure that globally we're being as inclusive as possible. We have an online forum. We've also had to, I don't know if any of you guys have used Trello for project management and organization, but they started charging us, which we didn't want to pay. So we switched to ClickUp. I don't think it's as good. If any of you have project management that is free and you would like to help out this single cell community, please let us know. Otherwise, if you're looking for a space, ClickUp's not too bad. And I believe that's our group. If you're interested, there's the link.
So everybody who is actually running a local small scale Galaxy installation knows that it's pretty different from one of the main Galaxy users. Most of us just run this as a hobby because you have a day job to do as well. Probably the biggest problem is lacking support from our local system admins or IT department. Then we try to connect this to local user authentication. We try to connect this to our local data. When we want to troubleshoot, it's difficult to talk to colleagues because our servers are behind some firewalls, behind whatever different logins. Then after doing all this work, we actually have a problem convincing our users to use our service. So we kind of came together as a group in order to identify problems we have in common, trying to help each other, and also within the Galaxy community to have a bigger voice of all those lonely sysadmin or these lonely Galaxy admins. We have a mailing list, so far 32 members. We have meetings every other month. We use for sort of short-term problems, we just use the admins matrix channel. We didn't want to sort of duplicate efforts here. Yeah. Thank you very much. And we have a boss at pretty much at the end of the conference.
Thanks. Yeah. So images. Yeah. The first tools for image analysis in Galaxy date back to the year of 2015, which is now almost 10 years ago. And it's the first time that Galaxy for microscopic image analysis was introduced to a wider audience. This was by the year of 2018 at the GCC of 2018. And it wasn't before the year of 2022 that the need for more collaboration between the contributors of the different tools was recognized. So the need to actually form a community. And although some of you might have heard of us as imaging people by now, actually at the Biohackathon Europe last year, those of us who attended agreed upon calling ourselves the image community. Yeah, by now we have fostered more than 100 tools for very different image analysis and image processing tasks, including segmentation, image registration, object tracking, and preprocessing. Just to line it out very briefly, segmentation is a task of delineating individual objects in images, such as cell nuclei in this example. Registration is a task of transforming different images into a single common coordinate system, and object tracking is the task of determining object trajectories of moving objects within temporal image sequences, and preprocessing usually involves tasks such as denoising, deblurring, or enhancing image contrast for subsequent analysis in other processing steps. So we organize mainly via the Euro Bioimaging Expert Group for Fair Image Data Workflows. We have monthly calls on third Wednesday at 4 p.m. Central European Time. We also have a Matrix channel. If you have any questions regarding image analysis in Galaxy or want to connect with us, feel free to join us.
Hi, so let's talk about genome annotation. Galaxy genome annotation, GGA, has been there since a few years, but now the context is that we have these huge sequencing projects under the Earth Biogenome projects, where you want to sequence all of Earth eukaryotic biodiversity, which is a quite big project. And in this kind of project, annotation is crucial to make sense of the genome sequence that you get after analysis, assembly. But the fact is, annotation is a bit of a mess because there is no currently unique gold standard method that can work on all genomes. And we even have problem to define what is a good annotation. So we are trying to work on tooling, workflows, visualization, and training for the genome annotation in Galaxy. We also have worked on the manual curation infrastructure, which is based on Apollo, and it is deployed on usegalaxy.eu/Apollo. And we've worked also on integration of Galaxy with Gmod application with Python libraries and Docker images. And we also have a subdomain at usegalaxy.eu dedicated to annotation. And you have some nice pictures of all of these tools, workflow, visualization training at the bottom of the slide. And so we have one page that can be found at this URL, and we have a matrix channel where you can discuss genome annotation tools. And now we are mostly meeting using the EBP, ERGA, VGP, or other Elixir biodiversity community meetings that occur regularly and talk already about genome annotation. So you can join us quite easily. That's it for me.
I think it's the last community, so I will talk about microgalaxy. So in the aim of the almost 50 people joining in the mailing list. So why do we exist? So we are interested in microbiology research, and there is really an increasing need for programming skills, specialized styles, and it needs a lot of computational power to run that. So the missions of the community is to develop and sustain microbiome data analysis in Galaxy, and microbiome is really large, so it includes also microbiome in there. And the aim is also to implement best practices and standardize them, expand the documentation and training, coordinate the efforts to avoid redundancy in work, in tools, workflows, trainings, share all the work we are doing together inside Galaxy, but also outside Galaxy, really making an effort on outreaching everyone, and support each other in that. So what we have been up to, so we have in the microgalaxy, at least in Galaxy world, more than 260 tool suites available for microbiome data analysis, more than 50 public workflows, not all available in IWC, but we are working on that. We have more than 30 GTN tutorials and learning pathway available. And we really worked lately on improving the annotations of all these resources within the Galaxy ecosystem. So we worked a lot with the biodot tool, so Elixir registry, to improve the that metadata available there. We linked a lot of resources to biodot tools, and we annotated tutorials with EDAMs. So work in progress, we aim to offer now 2 tracks during the Galaxy Training Academies, and we are developing the Galaxy Codex, so I will have a talk tomorrow about that. And we are writing 2 papers about all the effort we are doing within the Galaxy ecosystems for supporting microbiome data analysis. And we have subdomains available on Use Galaxy EU and so on in other servers. How do we function? So we have quarterly 2 hours meetings. We try to alternate between time zones. We have 2 working groups working on tools and the papers, and we have a page on the hub, we have a chat, we have a mailing list, we have Google Calendar if you want to follow everything that is happening. And we have a BOF tonight. Just a pity that it's in the same time as your BOF, because we need to discuss together at some point. Yeah, good, that's all on my side.
All right. I'm going to start by talking a little bit about some pre-24.1 stuff. We added the ability for users in Galaxy to pick where data sets are being stored. So admins can set up multiple different object stores and users can make choices. I'm going to go really quick through this because I briefly talked about it at a previous GCC, but we implemented a visual language for sort of summarizing these object stores. And we gave users a lot of power in terms of specifying at the tool level or the workflow level where the data will be stored. And then also you can specify history default or user-wide default. The sort of initial use case that drove some of this work is this very common request we get on usegalaxy.org that we need more space to do research. Can I have a small short-term increase? I woke up this morning in Galaxy Help that the top message was this. I need extra space. I'm willing to pay. So we think a lot of this can just be done with enabling like a short-term scratch storage on usegalaxy.org. So that's available today. Huge thanks to Marius, Nate, and Martin for helping enable this. But you know, the storage quota is 4 times as large as the default quota when you log in. Everything is deleted every 30 days, and it's available to everyone today. So that's pretty exciting. It's always a little. It took a while between like implementing object store selection and getting it into users' hands, and so having users now is a little bit stressful because people are using it. But it has pushed to make some other changes to object stores. So we're working to integrate it into the storage management feature and allowing users to switch. You know, after the fact, maybe run analysis in short-term storage and then switch it to long-term storage or just the outputs or whatever. So we've enabled these changes, and we did a bunch of cool things to sort of make it more production ready prior to 24.1. All that's cool, but again, like this message says, I should just be able to pay, right? There's no technical reason to prevent users from attaching their own cloud storage on something like usegalaxy.org. And so that 24.1 enables this this feature, letting users bring their own storage, and we call it object store templates and file source templates. I'm making a slight distinction here between files and datasets. I'm not. Galaxy makes a slight distinction here. Files are just, files, the way you think about them. The datasets in Galaxy have key value metadata, have maybe BAM indices, are there sort of index data, maybe extra files, maybe you have an HTML file and associated images. And so we have two concepts here. We have file sources and we have object stores. And there are different ways to sort of think about data inside of Galaxy and data outside of Galaxy. But the features I'm going to talk about work pretty uniformly across both of these. Previously, the configuration for both of these kinds of objects were global things, but we've seen this in several talks already today. Like users need to be able to configure their own storage for their own needs. It was mentioned in the keynote. It was and it was mentioned in the other work that's happening here in Brno. Hopefully these are the features that will enable those sort of use cases. In 24.1, if your admin has configured these templates, your user preferences will now let you manage storage locations. So storage locations is what the user-facing version of what we call an object store. Martin decided that language was a little obtuse. I'm still using object store because I'm a backend programmer, but storage location is how users will refer to them. And then the remote file sources, that language is the same. Anyway, the template, it defines a user interface for providing details about the storage and then describes how to connect that storage, how to describes the configuration that Galaxy would use for those storages. And those configurations are the same things that an admin would do. And so I was thinking about examples for this, and the sort of simplest possible template I can imagine that's production useful is allowing users to attach like publicly accessible S3 buckets as file sources. So if you open main or EU's file source configuration, there's a bunch of globally accessible buckets that are available to all users, but they're just like, it's the same piece of config over and over again, and it's just a different bucket name. And so this would be a comparable template that would let users describe their own their own bucket. The variables here describe, the things that the user is going to specify. The help is in Markdown. The description is in Markdown. And then the configuration there is exactly what you would see in the file source conf. That's exactly what the admin would configure. What the user sees is just this. It's just a nice form. And this one's pretty simple. If I create a file source for this, I give it a name and description. That's just for the user's own tracking. And then I just give it that bucket name that the template described. But there's a lot of complexity that's available in templates, and the existing templates that are offered in 24.1 have a lot more complexity. Another example here is a file source that's just any FTP server. And these production file sources that I'm going to talk about here, that they all have help. They have some descriptions. The FTP example, many FTP servers have passwords. And so part of this whole configuration process is how do we keep track of secrets? And I want to say a huge thanks to Nuan for developing this this vault abstraction in Galaxy for storing secrets. The templates build pretty heavily on that, and they provide a nice sort of refined user interface that ties those things together. But information on configuring the backend is there for that stuff, for the secret configuration. But how you use it in the actual template is very straightforward. And how the user sees it, again, is pretty straightforward. It looks like just another entry in the form. Like I said, 24.1 comes with a bunch of these production-ready file sources that admins can just link in for connecting to Azure generic S3 storage. So we saw a lot about Ceph today, right? This is, you could, if Brno wanted to, if it had a set of Ceph servers that had a certain parameterization, they could create templates that are specific to the institution, right? And so we have really detailed documentation for how to do that. But these ones are production ones that could sort of connect to any S3 service. So maybe it'd be a little over parameterized for the typical user, but despite the complexity of these templates, they do have documentation, like I mentioned. Object stores, this is where your data sets live in Galaxy. Again, a bunch of the sort of default cloud providers are available in 24.1. So where am I on time? 2 minutes, okay. Doing this required a bunch of, it didn't require, but I did a bunch of work on the back end to make all of the object stores more, all the cloud object stores more production ready. I saw S3 connected to Galaxy in a few different slides, and I was like, oh, I don't know that I trusted those object stores before, but we've solved a bunch of bugs. A bunch of long-standing issues have been resolved in 24.1, and I'm feeling much better about the whole concept of connecting to these kinds of storage services in Galaxy. And that's 24.1. Looking forward to 24.2 and what's coming with this feature. When we merged the Galaxy file sources, a motivating example was Dropbox. It was really cool. You could connect Galaxy to a Dropbox account with just a little user token. And Dropbox decided, maybe using an API key to complete access everything in your account is a problem. And so they quickly moved away from the API that made that pretty cool, and they transitioned to OAuth 2. And that's too bad for us, but OAuth 2 does provide a more secure service, and it can provide a better user experience because users don't need to worry about keys, and so in 24.2, I've extended the file source concept for these templates to enable OAuth 2 versions of these templates. And so for instance, the Dropbox one that's included in this open PR will just, it just takes the user to Dropbox and it says, give Galaxy permission, right? We're not even worried about keys at this point. So it should be more user-friendly than ever. It is a little bit harder for the admin to set up, so the PR does include detailed descriptions about how the admin can set up the keys and work with Dropbox to enable these things. Google Drive support is in that PR also. I have some initial thoughts on OneDrive that are not that interesting. And then more detailed user messages, and this is a sort of different realm for Galaxy. I think letting users describe technical resources is not something Galaxy has done in the past. So I think providing really detailed error messages is important. And so I've put more work than I've maybe done for other plugin frameworks into this feature, and those will be enabled in 24.2. All right, thanks. And any questions?
I love you, John.
I love you too, Bjorn.
So, I mean, honestly, this will take us busy for the next years to roll that out and to bring this actually to the users. So this is amazing work.
What can I do to help?
Yeah, that's a good question. I have a different question. So you also introduced the deferred data set stuff, I think a year or two back. So how is the connection with that? Can we enable, for example, a file source plugin to be by default deferred? So that if I include my Dropbox, all this data is by default ingested as deferred data set. Does that make sense? Is that something that is maybe already working or we should integrate?
In theory, you can mark any file source import as deferred. I had not considered making that sort of deferred by default an option, but I think it could make sense depending on the file source. I would like to see deferred files get more use cases, I'd like to see some user stories that say, yeah, we use deferred files, deferred data set evaluation, but how do we think about that feature, and we did something productive with it. But I think that's a good idea, and I can't imagine why it would not be possible to implement that feature. So sounds great. Thanks so much.
Yeah. Hi. So it's all going to be about workflows. So let's dive right in. And I decided I want to actually change that talk title because that would be a long talk, but let's get it concise. So I want to talk to you about the Intergalactic Workflow Commission, and I think you've heard the term being thrown around. And I want to make a pledge that please come and join us, either as somebody running the workflow or as someone authoring these workflows. So there's a role for everyone. Yeah. So what is actually the IWC? It's a group of volunteer workflow aficionados. So yes, it's a community place centered around Galaxy workflows. I think we've sort of always had this in mind for a long time. In 2018 in Portland, we came together and said, we're going to do this. And it only took another 3 years for the first workflow to appear. So super speed. And I mean, the goals are kind of to lower the barrier to workflow use. I think workflows are amazing. They provide so much context to an analysis. And for consumers of workflows, users, it is far easier to run a workflow than to set 51 parameters in like a mapping tool to make it appropriate to their particular analysis. So the goal is really share and reuse production-ready workflows. We also want to assist with workflow development and testing. Testing is really key. I'll come back to that later. We want to communicate the requirements to the development team. So, you know, some of the IWC members are on the development team, but also, you know, if there's a pressing need, we can like make some noise. And we want to document tips and tricks and contribute to the training material. All these little weird things to just, you know, that are possible, but maybe you don't know about. So where can you find IWC workflows? Maybe the most obvious one is if you, here's a screenshot of the new interface. So if you go to workflows, you have this little tabbed interface and you can click on public workflows, and on the Use Galaxy instances, so .org, .org.eu, and others we can easily add more. You find them automatically, and you can click on IWC to get all the IWC ones, or you can click on the tags. There will be a talk by Ali Reza showing all of that in detail. So another way to find them is through Docstore or Workflow Hub. What's nice about them is that they're organized into collections. So, you know, a workflow can appear in multiple collections. And, you know, this is great for findability. Yep, so you have your choice of where to get the workflow from. So at this point, I want to show you a little bit about the evolution of the IWC. So the first workflow and corresponding test infrastructure we've added in February 2021. And at that time, that was heavily driven by COVID-19 analysis workflows. If you don't know, we ran a ginormous amount of workflow invocations or workflow runs against COVID data to do some raw data analysis. And in 2021, we added 12 workflows. In 2022, we added another 10. In 2023, we added another 23. And in 2024, so this year, that's not over yet, we've already added 22 workflows. And there's another 10 that are almost ready. So that brings us to a total of 67 workflows. And you can see that like the, is there a pointer? Well, I mean, you can see that like the intensity of contribution increases. That's really what we want to see. And then we can also look at like what fraction of workflows are actually IWC workflows when they're being run. So in the blue bar, this is the total number of workflow runs per year on usegalaxy.org. So, and I've excluded the SARS-CoV-2 workflows because these made up like a huge fraction of it, and like we're sort of distorting the image a little bit. So we were like just below 60,000 and then going just above in 2021 to 2023. And we're consistently about 4 or 5% of runs. And I mean, this data is a little complicated, but like a good fraction of these workflows were IWC workflows. But I mean, I'm also hoping that in the future we can increase that percentage. And then how many users are actually using these workflows? Also a significant portion, but not quite as big as I would hope. And so that's why I'm telling you all of this. These are great workflows. Find them, use them. It does, however, show that the people that are running these workflows are running them a lot. And that's great. I mean, we want that. If a workflow is never being run, you'll never find the bugs. You'll never hear about all the improvements you could be doing.
So as a workflow author, why would you submit a workflow to the IWC? So we'll help with discoverability. As I mentioned, all those workflows are uploaded to different places, and you don't need to like worry about that for each and every single thing. You can get DOIs, hopefully citations from users that are running your workflows. We have daily workflow tests. So these are really super important because it allows us to confidently make changes and keep up to date with new tools, new bug fixes, all that stuff. It's really important to have tests. At the same time, we can also make sure that like we're not inadvertently breaking anything as we deploy new versions of the Galaxy software itself. Again, the tests enable us to do automatic updates. So we can look at what tool has been updated because like the tool developer made a new version and we can update the workflow automatically. They'll open the pull request, the tests run. And then, you know, if the tests are green and everything looks good and there was no breaking change, we can like confidently merge it. And so then users will always find the latest version of the workflow with the latest version of the tools. So always at the state of the art. Everything is nicely versioned. So, you know, if for your big project, you've been running version, 2 of the workflow, and you want to keep doing that, you can. If you want to get the latest one, you can also do that. Hopefully you'll be getting bug reports, contributions from users. And so if you're saying, I want in, how do I join? We have a link here to add yourself to the list of members. You can join our chat. We have a video call every second Thursday. We'll be working on workflows during the CoFest. There'll be an advanced workflow development training tomorrow from 4 to 6. And really any contribution is welcome. Questions, bug reports, writing or teaching tutorials would be amazing. Small or large changes, everything welcome, new workflows. And please tell your friends, there is this project, they have great workflows. If you need to be doing something, check if there's maybe one that already fits the bill. And so I want to leave you with the vision for the future. I think workflows should be the primary entry point for analysis for users. It is so much easier to run workflows than tools. We can provide so much more context for a given analysis, and showing the progress overall is so much simpler. And then I want to see a large collection of workflows that's ready to use. We want to make it easier to submit workflows to the IWC, and we want to have a lively, open, and collaborative community. So with that, I want to thank everyone that has contributed, all the developers that have been working on features. Yeah, thank you for your attention.
See, Alex is the closest.
So as you pointed out, a lot of the users who are running IWC workflows are running them a lot, which is showing that people really enjoy and like the outputs there. How are you planning on getting it in front of more users to make them more aware of IWC workflows?
Yeah, so I think it's important to have be part of trainings so that each training, maybe, we have these great tutorials, like we could also say like, hey, there's maybe an associated production ready workflow, have them be on the front page. A lot of like thematic Galaxy instances have like workflows that are specific to that instance. So like maybe have them really be on the front page and like really, it's also up to you, like use them and like, if you like them, recommend them to your colleagues. If you don't like them, let us know what you didn't like.
Thanks, Marius. Really nice summary. I wonder, maybe I missed it in your graphs. What fraction of tools are being executed outside of workflows versus inside? Yeah, I think there's an intuition that there's a lot that people are just clicking and running tools tool by tool.
Yeah,
an easy way to gather that data.
I mean, we could look into that. I haven't done that, so it's a bit hard to guess. I don't know. Maybe Nate knows the total number of jobs we run per year. I don't know if you know that off the top of your head. Okay, hundreds of thousands per month. I'm gonna say like in a couple of million if we get like 100,000 executions. No, maybe 50-50. It really depends. Certainly if we count in like the large projects that run things automated through bots, yeah, we might be there. I don't really know.
Great, thanks.
Talking about the driving decisions with data and using this new dashboard called key usage indicators to to inspect some of the data. We can't answer that question today in this dashboard I'm going to present, but we're pretty close to being able to answer it. So data-driven decision-making has remained, I guess, for a number of years now, sort of the most definitive method to work for driving good decisions, right? And you may not use these companies, the logos that I've shown up here, but I'll bet you know all of them, and you know all of them because they've helped a lot of people. So my thinking is, how can we as Galaxy help more people? And that starts by driving some decisions from data rather than hunches and sort of being limited to just the users that interact. And because we have a lot more users that are actually using our services and users that we come in touch with. So, but of course, we're all about data, I guess. So data is not using data, I guess, is not a new thing in the Galaxy world. System administrators, in particular, have used data for a number of years to monitor their services, right? We have Grafana dashboards for all the big used Galaxy servers. I'm sure the small admins have some as well. And these things show things like how many jobs do we have in a queue, how many jobs are run or any nodes up or down, a lot of stuff, right? We also have Sentry as a way to track bugs, which recently has taken a big focus at usegalaxy.org to squash as many of them as we can. People that are working on it more so than me, of course. And it's proven to really work and sort of reinforces this idea that data can help us deliver a better service. However, Grafana itself is a wonderful tool, very powerful, and that power has been exercised by the admins, the devs, and they've created a number of dashboards that range from sort of development mode to very complex ones. And they tend, but the problem is that they tend to be difficult to sort of reason if you're not a developer or somebody that stares at the dashboard at all times. Meanwhile, a lot of times, when we come to Galaxy, we come to a new project. So, for other people, where Galaxy is a new project, you want like a high-level picture. If you're a PI and you want to report what the service that you're getting funded for is doing, how it's doing, you want this high-level report. And the Freiburg Galaxy team does this annual report. It's wonderful, right? It's got many pages on there of text explaining stuff, but it also has this table or this figure that sort of captures where Galaxy stands today in terms of, in a digestible format, in terms of the numbers that represent its use. And so that's kind of what the idea basically emerged for this. It's let's digest the data that's in Grafana and put it into a digestible format. And so that's where this key usage indicators dashboard came up, and that's what it is today. It's a simple dashboard that just captures key usage indicators of Galaxy servers. You can go to it at gxy.io/KUI and see data for user registrations, user engagement, service usage data, and job data in those categories, and each has a couple of plots sort of detailing it. And can get into the details a bit in a second, but these are the kind of the high-level concepts. And so, as one example, I guess, of what we can do here is get up-to-date information, right? Many of us give a lot of talks throughout the year, and sometimes you hear we've run 600,000 jobs last month. Sometimes you hear we've run 1 million jobs, and so there's some discrepancy, I guess, in that. So here you can go to this dashboard at any point in time and get the accurate, up-to-date information to see where our services stand. In addition to points, data points in time, we can observe trends. Right. So if we look at the number of jobs run over a period of time, we see that for example, people like to take summer holidays, spend some time with the family around the Christmas and New Year's period, and so again, if you're an admin and you're planning a big upgrade, maybe it's worthwhile considering let's do it when there's less usage than more, and so being able to see such trends. And then we can also look at some details, right? And if we just look at last month, most recent one, May 24 and May 23, we see there's a almost 300,000 job drop in the number of jobs. And so, it makes you wonder, like, well, what happened? Is our usage dropping or not? And so we scroll around a bit more on the dashboard and we'll look at what else can we see here. And we can see that most jobs in May 23 were run by new users as opposed to existing users that have used the service for more than a given month. And so that, okay, gives us another lead to see where we are. And then you got to go off the dashboard a little bit, look through our documentation and see that last year we had the smorgasbord held in May that brought 2,000 users or so to the Use Galaxy servers, which is sort of an explanation of this particular data point, right? And as a pitch, so it's not going to be called the smorgasbord, but in October this year we have the Galaxy Training Academy, which is the equivalent of the past year's smorgasbord. So share the world, and I mean share the information with the world, please. So what else can we do, right? So all these dashboards that we've seen to date are isolated to one server. Here we have data that crosses the server. So all Use Galaxy servers have agreed to sort of contribute the usage data to this dashboard, and now we can use this data to compare and contrast how the given services are running. And it's not a competition; it just gives us a global a state of sort of global impact, of global usage that our services offer to the world, right? So and again, that's sort of an avenue of growth for us, has been, right? And so I think to date, I certainly am excited about the idea that I can always get up-to-date information from this dashboard. I can observe some trends. And then I always think we should celebrate milestones, right? I just learned yesterday that Australians celebrate every 1 millionth job. They write up a little story about the user that ran that millionth job, and it's a great way to sort of empower the user, celebrate the achievement within the team, and so it's a good way to again build culture. But there's also kind of the future view in that this is just the first stab, right? This is the first time this is being presented. It hasn't really been used to make a decision on governance or anything of such kind. But it does give us some options to think about, how can we enhance our onboarding or retention of users, again, based on these data points. We can talk about feature development based on what people are or aren't necessarily using. We just saw some plots about the workflow usage as an example, and we can just dig a little bit deeper into that. And so that's where we are today. It doesn't mean that's where we have to be. The technology choices, the plots that are on that dashboard are all up for debate. It's whatever's useful to the people that we can put up. So I have a poster this afternoon with all of these details, and if you want to chat about that, if you want to talk more about the details of where can this go and want to contribute data to it as well, come to the BOF also this afternoon. So with that, thank you. And there's a question, I guess I can take one. Alrighty, pass. Forget who's next.
Thank you. That was really cool. So the ingest is mostly coming from like database queries, or?
So so there's the queries are in GXadmin, and queries get run monthly by a cron job, deposited into BigQuery, and then visualized in this Looker Studio. Again, technology choices were such, but they don't have to remain such forever.
Yeah, I was wondering if we can also put in some of the Grafana stats, because that's like not open everywhere, and like it's varying quality sometimes. So maybe if we put that in there, we get some additional cool things.
Yeah, absolutely. Thanks.
Thanks. I'm curious about, so you've gathered all this data. It's really giving us some insight that like what we measure, I think really matters in terms of which way we go. And that decision making seems like if you measure the number of new users, and that nice illustration you gave of like, oh, there was a smorgasbord that previous time, so it kind of threw off the numbers. I'm wondering if you have any thoughts about sort of metrics that are robust to those kind of event-based things that like, what are the key things that tell us about health of Galaxy going forward? Like that might give us a clue about, are people really using Galaxy to do real work? Maybe that's an important question to ask.
Yeah. I mean, I'd like to say we have the data that we need, but I'm afraid we don't necessarily, right? I'd love to get some more data on the demographics of the users. You know, what's their role, for example, in life, whether they're a student, whether they're a researcher, whether they're a biologist or a technical, you know, staff member. And I think that would inspire us, I guess, to interpret some of this data better, right? So when I put these plots together, I tried to pull out data that gives us some answers, right? How many jobs we run in a month it's an interesting value, but it's not particularly useful whether our people are doing good, cool things or whether they're just running a bunch of jobs, you know, because these plots don't exclude bot jobs, for example, right? So a lot of these could be bot jobs. So I don't know, if you look through the dashboard, I think there's a lot that that isn't based on the number of jobs, right? We have some data points that say, you know, what are our error rates, and they fluctuate. Like when you know, there are error rates by new users, by users that have been around for a while, and it tends to be that users that have been around for longer have higher error rates, for example, right? So it gives us an idea that they're doing more experimental stuff, and it could potentially mean that, you know, either our help needs to improve in that, you know, they're using wrong parameters, or maybe they're just doing things that you know, the data is not conducive to. But I don't know how we get to like a definitive answer, but there are some clues in there that can take us a bit closer to hopefully developing, you know, a feature enhancing this, well, hopefully the service. But thanks.
Hello, everyone. Nice to meet you. I'm Marcus from 52 North, which is a not-for-profit company full of research software engineers. And yeah, we cooperate with universities and with people who create consortia to apply for research proposals. And whenever they want to develop some software and they don't have the resources, they don't have the skills to do so, then they can contact us. And we join these projects as a work package, as a task or similar. And this is also what happened in the Aqua Infra project, which is about developing a research data infrastructure for marine and inland waters. So the problem is a bit that we have marine data and we have freshwater data, and the two are not very well connected. And the problem is not just that the two are not very well connected, but also in each of them, the data sets are not very well connected when it comes to national borders, when it comes to research disciplines, or also regarding research data infrastructures. And this is why we want to develop yet another research data infrastructure, which is not similar to the ones that we want to connect, because we just want to connect the existing data infrastructures, the data providers. It's not that we want to have another repository which contains all the data sets. This is not what we want to achieve. In the project, we have a couple of case studies. So we have the North Sea case study, we have the Baltic Sea case study, we have the Mediterranean region case study, and the Pan-European case study. And just imagine that they are all doing the research in these areas. They are using the data that is already available. But the problem is that the data sets are, as I said, not very well connected. And this is where our research data infrastructure comes into play. So what we would like to do is to connect these data sets and provide them in a ready-to-use form so that the researchers can use them in an analysis environment, just like Galaxy. So the overall objective is to create some kind of a virtual research data environment, which is at the moment Galaxy. And we want to have fair multidisciplinary data. I think that the slides are not very current. Is that possible? I corrected the slides this morning. No, they are not the. To maybe refresh them.
Did you change the link though?
No. Yeah, this looks better. Yeah, that's fine. Thanks. Great. So we have some objectives that are copied from the proposal, and I don't want to go into detail in each of them, but I just wanted to show that there are quite a few buzzwords that we mentioned in the proposal. And yeah, I just wanted to show that we cover quite a few aspects that are relevant in open science and also at different stages of the research cycle. So it's about data collection, it's about data analysis, It's about publication of data and also analysis scripts, workflows, and stuff like that. It's also about publishing these materials in permanent repositories. And this is what our infrastructure should achieve at the end. This is an overview of the key components that we have in our platform. So we have the data discovery and access service, in short DDAS, which is the backend service in our project. But I'm not in charge for that, or my group is not in charge for that. We are working on the Aqua Infra interaction platform in the virtual research environment. So the Aqua Infra interaction platform is based on the DDAS, which is it's some kind of a user interface for discovering resources. And from there, they should get to the virtual research environment, which is in our case Galaxy at the moment. And the virtual research environment can either import the resources from the AIP, from the interaction platform, or from the data discovery and access service. So we have basically on the front end side, we have two main concepts, which is the Aqua Infra interaction platform and the virtual research environment. The AIP, the interaction platform, is more about searching for resources. It's about accessing these resources and getting a very initial understanding what the resources are about. It's not so much about analyzing the resources. This is what happens in the virtual research environment, which is done using Galaxy. And there it's more about reproducing results using workflows. It's about developing new analysis and publishing and sharing these new outputs. And the overarching goal is, I also copied that from the proposal because I actually find it quite nice to formulate it like that. It's about transforming the way researchers, as well as the public and private sectors, to create, share, and exploit research outputs. So data, publications, protocols, methodologies, software code, and so on. So the idea of the project is to demonstrate the benefit of fair data, of open data, reproducible research, and so on. This is the architecture. I will not go into detail because I have a very nice poster that you can visit later, and then we can discuss the architecture in more detail. I just wanted to show in this picture that we don't just have the data discovery and access service, not just the AIP and the VRE. We are also using external components like Zenodo. So the very nice plugin that was developed by David is already in use. So it came at a perfect time because we have a deliverable now, and I was able to link to that already now, so this was very great. So yeah, we are using Zenodo to also publish the outputs. This is how the platform looks like. So it starts with a very simple UI to search for resources. And on the next page, you can search for the different data providers on the right side. So each of them hosts data and sends metadata to the DDAS, and we search the DDAS for the data sets. You can also set a bounding box to restrict the search results with spatial properties. And yeah, of course, you can also enter a search term. And if you click on one of the search results, then you see this kind of details page, which shows the metadata of every resource. And on the right side, you can see the external resources. And the second button is about importing the data to Galaxy. And this is what we did with this data source tool in Galaxy. And this is some kind of a bidirectional connection between the AIP and Galaxy. So from this page, you can get to Galaxy, and Galaxy directly downloads the data. And this is only available if there's also a link in the metadata to the data. And this is unfortunately not always the case. So many resources have metadata, but the download link is not always in the metadata. So we have a bit of a problem how to handle resources where the download link is missing. But Galaxy, so we have an own subdomain, aquausegalaxy.eu, and thank you. And there we also have the Aqua Infra importer, which redirects you back to the AIP, where you can search for resources and then import those resources to the virtual research environment in Galaxy. So this is the bidirectional connection between the two components. So what's next? So we have these use cases. We have the researchers that are working in these use cases. They are developing their analysis, their scripts in R and Python and whatever. And our goal is now to take these scripts, to break them down into single functions and import them as tools in Galaxy so that they can create some workflows and share them with the public. All right, if you want to learn more, come to my poster or visit the website. there you will find more information, and these are the partners that I also want to acknowledge. And that's it from my side. Thanks for your attention.
Well, we may have a question for you, Marcus. Don't run just quite yet.
Just yeah, thank you for the talk. Just a very quick question. Maybe I missed it. So you showed that you can basically display the metadata, but can you also filter by the metadata immunity on this database? Because I'm asking, you have like initiatives like Track Otara, so like where they went with the boats from Emble around coastlines around Europe. So it would be actually beneficial to see for that particular data set, just pick me this data which is from UK or whatever. Is that possible?
It should be possible at some point in the future. So this is not what is existing already now, but we want to, or at least the backend team wants to develop a service where you can make such requests. That you can filter by bounding. Well, by bounding box is already possible, but that you can make some more exhaustive queries to the data set and get some specific subsets of the data set.
Okay, thank you.
Sergey.
Okay, yeah, I'm Sergey Kuba from Oak Ridge National Lab. Let me start with a couple of slides about our laboratory. It's the largest multidisciplinary laboratory in the United States, around 6,000 researchers working there, basically looking at high-performance computing, cybersecurity, fusion energy, biosciences, environmental scientists, neutron scientists, all kind of science, quantum computing, and many other things. And our group is collaborating tightly with neutron scientists. The laboratory has 2 experimental facilities on site. It's high-flux isotope reactor installation, neutron source. They have around 30 different beamlines and instruments to analyze the material, to look inside the material. And we are helping them. I'm from application engineering group, computational science and mathematics division. We are helping them to develop a platform for neutron scattering data analysis. So started around 2 years ago, and we came to the GCC 2024. Very optimistic, claiming that we want to run Galaxy on Frontier. That was and still is a supercomputer #1. Somehow that did not work out at that time. Frontier was not ready for us, but the year later we were able to run Galaxy on cross-facility, do cross-facility simulation, starting from experimental analysis cluster and then running HPC jobs on Summit, still not on Frontier, and then running some other tools or part of workflows on the Gates Cloud. That's our cloud instance. So this year, at the moment, we have around 60, 70 neutron tools, 6 workflows. They can run on different configurations, CPU, GPU, using Docker, Singularity, and host written in different languages, using OpenMP, MPI, or CUDA. Can run locally in the cloud, and finally they can run on Frontier, and Frontier is still supercomputer #1. Not for a long, I guess, but yeah, we are glad that it worked. So what do we do now? As it turned out, our users, they like interactive tools very much. Although we don't like them very much because we lose all this reproducibility and all the stuff, and user just open something and do something, and we don't know what, but it's kind of what they want. So we started with Jupyter notebooks. It was pretty easy. Then we implemented a couple of other tools. This one is actually a Windows application that runs an interactive tool, and people are happy they can do the crystallography analysis in those tools. Another example, that's a commercial software actually that requires a license server. We also created an interactive tool out of it. So now we need to solve the problem how to restrict user access to this or limit to specific users. But I think that's available in Galaxy. Just need to implement it. And basically, you can run any application. You have Linux desktop using FinLink client, so that you can install whatever you want, what our user want, and run it there through Galaxy. Well, started getting user feedback. It was kind of contradictional. Some of them, they like Galaxy. It's good to use for some cases, like tool development or running simple tools. But from the other side, also feedback that generic interfaces too complicated. You have tools with complex inputs when you have 50, 100 parameters being inputting them using the Galaxy interface was not very simple. They want to visualize the results more easily, not for visualization plug-in, and then visualize, select some region and reuse this as an input for the tool. That's a problematic, but yeah. But in general, yeah, interface is too complicated. The user come to facility, they have a couple days to do the experiment. They don't have a week or so to learn on the Galaxy. So we are trying to make things more simple for the user or more customized, if you wish. It's how it looks currently for the user. Kind of stripped Galaxy interface. It's completely new interface, basically. So you have this landing page where you only have a couple of tools that you need for your work. And different techniques, they want different landing page, so they don't know intersection, they don't have to select which tool they want. So we have this. And from this page, you can launch the more complicated tools. That's still Galaxy tools, still interactive tools. But now it's kind of looks like a web application, and you have your complicated input there. You have different tabs. You can input all your parameters or read them from config file. And from the same application, you can run jobs that then goes to Galaxy and runs there. And you can visualize your results in this application, or rerun and all this. So we're kind of creating separate application, customized and fine-tuned for specific use cases. But they all are interactive tools in Galaxy. So the technologies, as I said, they be launching using Galaxy. It integrates with workflows and the tools, the real data analysis and data reduction tools that we have in Galaxy. We can allocate specific resource for Galaxy based on demand. We can send this job on Frontier if you want, on a GPU virtual machine, wherever. And applications themselves are written in Tram. That's basically Python framework to create web application with Vue UTF-8 behind it. Since it's Python, it's relatively simple to write web application. You get this seamless data transfer between client and server. It's important, this remote and local rendering using VTK library. Just a couple of examples. VTK is pretty well-known library for 3D visualization. ParaView, if you know, it's using it. And it's the same company who did ParaView, basically. You can use Matplot and Plotly to use for charting. And of course, you can use Bible and what we use, because it's Python, to communicate with Galaxy. That's basically it. Couple examples. This one, single crystal visualization. You have all your input parameters composed as the user wants to see it. And then you can also have 3D visualization that can run on GPU or on some virtual machine. Yeah, have your structure of your crystal. Then you can click, select some data. And say, I want to run next tool from this selection, for example. And another tool for data reduction. They all look pretty similar, pretty still work in progress. We just hired a front web developer to make them look nicer. But that's basically the idea. And that's it.
Thank you, Sergey. Do we have any questions for Sergey?
Thank you. That was awesome as usual. I'm wondering, with the little web apps that you're writing, are you targeting the tools API or are you targeting the workflows API?
Both, actually. Some applications, they're just running single tool, some they run workflows.
Okay, and do you find like one or the other more enjoyable to program against?
It's difficult to figure out which inputs the tool and workflow needs. That's a pain. But once you figure that out, that's not a problem. One issue with workflow, this is as opposed to the tool, you don't really have workflow in your panel. So to run a workflow, user has to import this workflow from the shared workflows or something. As far as I understand, maybe I'm wrong. And a tool, you can just run a tool and that will work.
We can talk about it. I mean, it's possible too. But yeah.
Is it? Okay, we should talk about it.
Yeah. One more. There we go, Martin.
Thanks, Sergey. Awesome talk. You mentioned that one of the problems you're having with Galaxy is that your users are using complex inputs. Could you describe a little bit what they need or what they want to see?
Well, in Galaxy, you have kind of, if the input has multiple parameters, like I don't know, 20 or 50 parameters, then you have all of them on a single page, one after another. You cannot choose the layout, mainly how these inputs would be configured, like different tabs, configure different levels and all this stuff. That's the problem. How we solved it before in Galaxy, we just said use configuration JSON file that you prepare elsewhere, and that worked, but that's not the ideal solution.
Got it. Thank you.
We have Sam.
I think there is an option which might help a little bit to remedy that. There are like sections.
We use section, but it's still long.
They can collapse, or maybe conditionals, but okay. I just want to make sure that you tried those.
Yes, we tried everything.
Okay, thank you.
We have an online question too. I'll read it because we don't have other opportunities for interaction. So could we imagine a very simple interactive tool, a web page with a button where the user chooses whether to continue the workflow or stop it after manually reviewing current results? So a simple web page with a button where user chooses whether to continue on the workflow or stop it.
Yes, of course. Yeah, we actually implemented to Galaxy an option to stop the running job, which was not there before, but it's not push. So in Galaxy, you can either cancel job or wait until it's finished. We implemented, you can stop it and it will be green. You can still get the results, but we did not do the restart yet. But for the web application, you can do everything basically because it's customized and not generic. It's difficult to do such things generic, but easier.
Thank you.
So I'm talking here in the behalf of the Abromic consortiums. So it's a project I joined in January. And Abromix is the idea to develop a Galaxy-based One Health antimicrobial resistance platform. So why antimicrobial resistance? So antimicrobial resistance are bacteria, viruses, fungi, or parasites that no longer respond to antimicrobial medicines. And how antibiotic resistance happen is if you have a lot of germs, some can be drug resistance. The antibiotics kill all the bacteria except the ones that are resistant. And then the drug resistance bacteria can grow and take over. And then they can give also their resistance to other bacteria. Why is it important? Because it's one of the top global public health and development threats. It's around 1.27 million deaths per year due to antimicrobial resistance. It's like one death every 25 seconds. And so it's a really high cost every year. And the main bacteria involved are, for example, Escherichia coli that a lot of you have already heard about, but there is a lot of bacteria that are associated with resistance and attribute resistance. So the rationale behind the Abromix project was to there is an increasing access to sequencing for clinical laboratories, and there is an improvement of genomics, metagenomics that helps a lot to improve our knowledge about how antimicrobial resistance bacteria circulate and how mobile genetic elements and antibiotics resistant genes are spread. But most of the investigations are mostly siloed, while we really need a more global pictures there to help solving the problem there. And so to address this gap in the French ecosystem, so the Bromix project has been funded for 4 years to the idea to build a multi-omic One Health platforms for collaborations, data sharings between researcher clinicians, really for AMR surveillance. So the consortiums, it's a big consortium. It's 43 partners all over the French ecosystem in France. It's a multidisciplinary consortiums, including the national reference centers for antibiotic resistance. The coordination is made mostly by Claudine Meding at the French Institute of Bioinformatics. Sorry, it's really hard today. And the Institut Pasteur with Philippe Glaser. So the objectives are Bromix is to provide an open access platforms to genomics and metagenomic analysis with standardized and controlled pipelines and up-to-date set of different mathematicals, but also bioinformatics tools. The idea is also to build a repository of structured, interoperable, and standardized and well-annotated multi-omic biological data from human, mainly, but also animals and environmental, so especially wastewater. The idea is that it should be easy to use by non-bioinformaticians and experts in AMR analysis. And the idea is also to provide a shared platform to improve the surveillance and research on antibiotic resistance, again, across humans, animals, and environmental sector. So it's really an online One Health community-driven platform to scale up and improve surveillance of antibiotic resistance. So the program is developed in 6 work package. I will not go in details, but I think what is more interesting is the different use cases that will be covered. It's that we will cover multi-drug resistance bacteria, One Health approach, but also meta-pangenomics and aquatic environment, proteomics, so different types of omics there. How is the Bromix platform built? So on the top, the users, so the clinical microbiologists or the researchers provide their sequencing data, their worries and some sequencing information together with a bunch of metadata. So Julie here picture made a huge work on developing a referentials for metadata. Once the data, they have everything ready, they can submit everything through the Bromix web applications. So that has been developed by these people there and how it works. So don't forget what is on the left side is you have a project management aspect. So you submit your data through a project where you can have several people contributing to the same project. Then they upload their data, metadata. There is some validations for that. Then the analysis has been run. And then once the analysis is finished, they can visualize the result through the interface, and they have several analysis reports for quality metrics, analysis summary, the metadata summary, but also antimicrobial resistance analysis things. And then everything can be explored together, so globally, through the Bromix database. And how the jobs are run, everything, so all the data are analyzed via the use Galaxy FR servers, and with different workflows, so developed mostly by Pierre and Clea there. And all the tools needed for that has been added or updated on IUC, and the workflow has been added last week on IWC. And the connection between the platforms and the servers is done through Django to Galaxy that have been developed by Kenzo, Hugo, and Fabien in Pasteur. And everything runs on the French bioinformatics core cluster, thanks to Gilda somewhere hiding there. So the genomics workflow, we have currently we focus on the genomic aspect. So we have either short reads and or long reads available. So to the workflows. And the first idea is we have a sub-workflow for quality and contamination control, then workflows for assembly of the data, an annotation a general annotation with BACTA, but we try to also identify plasmids that are play a big role in antimicrobial resistance spread. IsoScans is for insertions, sequences, and integrant finder for integrants. And then we do steps of IMAR gene detection within using aggregate IMAR finder plus and star IMAR. So this workflow and all these sub-workflows are now available on Workflow Hub and Docstores via IWC, and they are all documented as a learning pathway on the GTN that you can find directly on the GTN. Yes, I'm almost there. So the next steps is to go beyond genomics. So first, still in genomics, we will add steps on core genome MLST. So it's currently working on that. We have a big steps on metagenomics. So there is a big work, groups of people working on data integrations and with different type of metagenomics. So we are planning to develop assembly-based approach, but gene catalog-based approach. So everything is under development right now. Pangenomics and multiomics will be also the next steps there. So you can come and visit the posters. I have a posters there if you are want to know more about that. And yeah, thank you for your attention.
Question already.
Thank you, Berenice. Very exciting project. Just a question of interest. So are these all disease cases? So it's basically where the patients developed already resistance, or are there also cases like where they study how nutrition impacts basically the antibiotic?
Not now. Currently, we are focusing on surveillance. So surveillance of the IMR and the spread of IMR. So really focusing on surveillance for now. And then once we have achieved that, we have enough data, then we can go to the next steps of what can be done afterwards.
Okay, thank you.
We have one more question.
No question. Everybody wants to go for lunch, I think. No, Martin.
Thanks, Bernice. Awesome talk. So based on all the data that you processed, are you like showing somewhere, visualizing the results, or like how these resistant bacteria spread, or like how it goes around?
So currently, no, it's not the case. Currently, you have only a map where you find all the samples that has been sequenced and analyzed, but not how it spread. But we first need to have data for that to achieve that. So we need first to get to that where data are submitted there, and then we can go next to more, long-term, this type of questions then.
Great, thank you.
All right, with that, we are going to close this morning's session. Thank Bernice again